That's the problem. There we go. And so we should be live. I'm just waiting for the broadcast to come up. Where's XSplit? I think it's a come up. Where's XSplit? I think it's a come up. Okay, grand. Well, are we on? I think that's a sign that we're on. Yeah, I can see us. Welcome everybody to this first Trolling with Logic of 2015. It's a very special 3-hour show. With us is Mortimer. How's your Christmas?
Oh, the Christmas was okay, but then I got the flu. So, yeah, I feel kind of like crap right now.
That seems to be going around a lot. And back on Ziller, welcome.
Hello.
And how's your Christmas?
I was okay, but I think Mortimer's flew over Christmas, so I've probably given it to you for the new year. Sorry, man.
And a special guest for this hour is my supervisor, Dr. Enrico Marsili.
Hi.
And so this is a 3-hour show. The first hour will be just myself and Enrico will be discussing the project that is on the experiment.com page and be answering questions. And then the second hour we have Professor Philip Moriarty back on the show once again to discuss science funding. And then the third one is actually, I'm surprised we actually were finally able to get Myles Power from the League of Nerds. And Zeller is very overexcited to talk to him, as am I. I'm actually quite a fan of his. So, sorry, I'm still suffering from the Christmas fatigue. So, well, maybe we should start about the actual project itself. The project we have is about producing metal. The project we have is about producing metal nanoparticles, and usually the conventional methods are either expensive or use potentially harmful compounds to make these nanoparticles. We take a biological method, which are cheaper, cleaner, and just easier to produce. However, there are problems associated with this type of production. Mainly that the nanoparticle production is very slow. It takes days to produce these nanoparticles, which adds to the cost, and the nanoparticles themselves have different sizes, which adds to the cost because the properties of nanoparticles are dependent on their size. So we hope to add focus to the nanoparticle population, to the nanoparticles used by the bacteria Shmonella, by adding in an electrical potential, and we hope to stimulate that bacteria using this electrical potential to increase the uniformity and decrease the amount of time needed. At the moment it takes about 100 minutes as opposed to 2 days, which is a great decrease in the time. Enrico, do you have anything to add to that? I'm just a little bit
Yes, no worry, I can take it for a few minutes from here. That certain metal-contaminated environment and sediments, including deep sea sediment, are able to interact directly with solid conducting surface like metals and electrodes. Basically, what's happening is that part of the energy respiration is channeled in oxidation of solid electron acceptor or donor, respectively. Shmonella onedensis is one of these bacteria, isolated originally from shallow lake sediment in the 80s, and is capable of interacting with a variety of surface and material that can use as either electron acceptor or electron donor. This is the general microbiological concept. There are various applications for this direct interaction. The first is a bioremediation, aka the bacteria is able to reduce or oxidize metal from its toxic to a less toxic form, or eventually to precipitate outside of the water phase. In other cases, the bacteria is able to perform anaerobic oxidation, which are very effective for detoxification of organic compounds, and that's one of the applications. That application, although it didn't reach the peak that we were expecting, is the production of electricity. In a device known as microbial fuel cells. The idea was to convert directly chemical energy from low-cost substrate like acetate, lactate, residual wastewater, and residual biomass into electrical energy. Although this device didn't scale up to the dimension required for mass production, it is still a very good device to investigate the nature of microbial metabolism and for some bioelectrosynthesis process. The other application, which stems from electrosynthesis in Shionella, is the production of nanoparticles. Nanoparticles have been produced for years in biological system, both viable cells and cells extract, because reducing enzyme either in vivo or in extract are able to interact directly with the metal ions and reduce them to the zero valent form, where they precipitate as nanoparticle or various form of aggregates. This is just fine. However, there are two problems associated with this biological reduction process and formation of nanoparticle. The first problem is the side distribution, which is very broad and is not useful for further application and for downstream processing. The second problem is the long time required for biosynthesis. Early experiment using fungal and bacterial biomass proved that using viable biomass, the reduction reaction occur in 24 to 72 hours, which is way too long for industrial application. Using using cell extract, the reaction time goes down up to 20 or 60 minutes. However, cell extract is expensive because it is not more viable cell, so it cannot replicate, and they need to be substituted every time. So the key idea is that bacteria are able to interact with electrodes. So we can probably provide a bit of extra energy in the form of applied potential to facilitate the metal reduction process, which occur either extracellularly or on the cell membrane. That's the idea that we started to explore with Michael in Singapore in 2012. We have currently some draft in preparation, and that's why we try to push down further this idea on the experiment.com platform and prepare a project that Michael is now supporting. Any other? Can you take it from here, Michael? And then.
Sure. The experiments to date so far, we've looked at the effect of the bacteria itself. We're looking at Shionella onidensis as compared to Shionella lohica. Shionella onidensis is actually the model organism at the moment, the model organism for bioelectricity, at least in terms of Shionella. But Shionella lohica actually has a faster metabolism. Therefore, we thought that it would be interesting to compare the two in terms of see if there's actually much of a difference in their performance for the nanoparticle production in the electrochemical cell. We're also looking at the electrode material, which can be an overlooked parameter.
Michael, sorry to interrupt you. Maybe we should outline better for the listeners how this process works. You have
yes, please, because I don't understand a damn thing.
Yes, good. Thanks for the feedback, guys. So, Michael, want to start describing how you set up on this experiment, and then tell basically which is the biological process happening, and then we go into further details.
Sure. Well, first of all, we set up a three electrode electrochemical cell. Now, there's as the name implies, there are three electrodes, obviously. The first one is the working electrode. This is where the magic happens, where we grow a biofilm and where we apply a set potential, for about 48 hours, and which allows the biofilm to form. It uses the actual working electrode, similar to how we use oxygen, as an electron acceptor. There's a counter electrode there that completes the circuit in the electrochemical cell, and there's a reference electrode. This allows the potentiostat, which is the controller of the system, to basically gauge it's a point of reference for the potentiostat that allows the working, so it can fix the potential of the working electrode.
Okay, but what is the biological process happening here? You want to talk about, or should I a moment?
The biological process, well, our hypothesis is at the moment is that once the biofilm is formed at the electrode, the working electrode, we can then charge the working electrode at a set potential for about 100 minutes or maybe even less. And basically the electrons, we assumed or hypothesized is that the electrons hop from the working electrode to this structure on the outer membrane on the bacterial cell surface, which then is transferred to the silver ions. Now we need to do a lot more experiments to actually demonstrate that this model is correct, and it may not be, but we're just at the very early stages of the actual mechanistic model. Maybe if there's anything else I might have missed out, or...
Is working for this with the system since 2 years and a half, so probably tend to assume a lot of things because it is the practical expert of the system. Let's back up a moment and try to make an analogy with the human respiration. Okay, we are all familiar with the process of respiration. Basically, we use oxygen. Sugar, glucose, and reduce oxygen to conserve the energy that in this process allow the maintenance of homeostasis and ultimately our life. Okay, I'm doing very simplistic summary here. Bacteria do something similar. They are able to respire either aerobically or anaerobically by reducing and transferring part of the electrons
you're cutting in and out there, Enrico.
The process of aerobic and anaerobic respiration, where oxidation of a substrate like glucose and acetate in bacteria result into the reduction of a terminal electron acceptor. Can be oxygen in case of aerobic respiration, metal or soluble redox acceptor in case of anaerobic respiration.
Sorry, you can kind
That again?
Yeah, sorry, but I can summarize just what you just said there.
Please.
For human cells, for usually for cells such as our cells in our body, we take glucose as sugar and convert that into energy through respiration. Now what happens is that we basically start to break down glucose, and which releases electrons, high energy electrons, which then can be used to form what is known as ATP, which is an energy carrier. But through this, you lose energy and give it to the ATP, and at the end we need to get rid of that electron. Otherwise a respiration will stop and we die. That's not good. So basically that is the role of oxygen. Oxygen comes into the body, comes into the cell to the mitochondria, and is then accepts the electron and hydrogens and 2 hydrogen ions to form water.
Okay. So what's happened then with bacteria, which is the analogy between bacterial respiration and cell respiration? That's where our starting point.
Yes. In the analogy with that transfers to bacteria as in an anaerobic environment. That means without oxygen, it obviously cannot use oxygen, so it's in trouble now. So basically, then has evolved this little ingenious little system where it can actually transfer these electrons to other electron acceptors other than oxygen, something we don't have that ability to do. So it can transfer it to metals such as iron oxides, gold, silver, even quite nasty little metals such as uranium and plutonium. This is what that Shrinella has done. Shrinella is not the only bacteria to do this. That's interesting enough. Geobacter can also do this process. However, we tended not to use Geobacter because if it's exposed to oxygen at all, it's going to die, which raises the cost of the whole synthesis and will basically raise the cost of our own lab, and it's quite finicky and not very appealing.
Okay, so which is the innovation of the project that you're proposing, Michael?
The innovation is that it combines the benefits of the biological methods, which are that the nanoparticle, the method is quite user-friendly. That means that each batch is more likely to be consistent, as opposed to processes which can be more complex, and then can lead to differences in the nanoparticles produced. The time is also drastically decreased. For Shrinella, it's 48 hours, 2 days, to 100 minutes, and sorry, there is one more. And the size distribution can be quite narrow, which is actually something we're working on at the moment to try to further decrease in order to match the conventional methods.
Okay, but why? You say that in your experiment project that I'm reading right now, even if I wrote with you, it's okay. You said that you are applying a potential, meaning electrochemical potential to these cells. Why is it important to do that, and which is the resulting innovation in this process?
The electrical potential actually is the stimulus. The electrical potential actually allows for an increase in the metal reduction rate. So that means that we're transferring electrons to the metal ions at a much faster rate. This means that the synthesis time decreases, and the size distribution of the nanoparticles will also decrease because of the increased focus.
So basically what's happening is that by applying a potential, we are providing an external contribution of energy.
Yes.
And since this is a catalytic reaction that results in the creation of a nanoparticle, we are facilitating this reaction by adding energy. This can on one side decrease the activation barrier needed for the reaction, which in the case of silver nanoparticle is fairly small. On the other side can increase the rate. Increasing the rate of nanoparticle formation has an interesting effect on the size distribution. We tend to have a smaller nanoparticle and created producing much faster. Instead, on the other hand, natural quote-unquote production of nanoparticle results in a wider size distribution and much slower process because the goal of bacteria is not to produce a nanoparticle but just to survive. So with potential, we are providing an additional help for this process, and more interestingly, by changing potential, we might in the future be able to determine the size distribution. So ideally, we should tune the potential applied to obtain the distribution that we want for our specific process, be it a drug delivery process or a nanomaterial production process.
Okay. And with nanoparticles, the smaller size is better, especially for nanoparticles like silver. Is there any questions from maybe our other hosts?
Well, I don't really know where to start because I haven't really understood all that much of this. It's basically what I guess I'm wondering about is, as I understood it, the nanoparticles were supposed to be used for in the medical treatments. Or is it just like you don't really care what they're used for, you just want to know how to make them? Or
At the moment, we're more concerned with the synthesis and then finding the application, because at the moment that's what my PhD is mainly concerned about. But there are applications for silver nanoparticles already being looked into, such as silver is a very good antimicrobial agent. Silver can be used for drug delivery and for catalytic purposes, in order to speed up slow down reactions. But silver is not the only metal we will be interested in, or if someone else takes on this project after I'm finished with it. Silver may not be the only metal that can be used in this process. We could also use other metals such as gold or palladium, which have their own applications. Gold is actually quite a fantastic metal because it's not toxic, but it's not toxic, but can have a wide range of applications, such as drug delivery. We can use it to deliver drugs in the body. You can use it in the treatment of tumors. It has applications in electronics, which I'm not really too familiar with, and wouldn't be too familiar with the electronic purposes of these gold nanoparticles. And it can also be used in sensors in order to detect bacterial DNA or tumor cells or other antigens.
Michael,
yeah.
Let me interject a moment.
Sure.
Going to the question of the drug delivery. When doing drug delivery, it's quite important to have a reproducible material, in this case nanoparticles, so that we attach our drug with a very well-defined geometry, and the drug kinetic is the same from batch to batch. This is usually obtained through a very complex standardization process in chemical nanoparticle production. This is probably the most expensive part, and we think we can save some of this hassle by producing nanoparticles with much more focused distribution using bacteria. So the main application will be drug delivery or direct application to biofilm, infection, cleaning of linen in the hospital, and so on. However, prior to this application, which are very interesting but are still far from us, we need to define, which you need to understand, which is the best way of producing a nanoparticle with good side distribution. And that's exactly what the PhD of Michael is about. Once we fix this part and we understand how to produce nanoparticle with the defined side distribution, we can then start investigating the application. And Michael is collaborating with Paul Cahill at the CU, which is much more interested in the downstream application, and will take the work from there.
Okay, I actually got a couple of questions from chat. This one from Microblogganazin. So you were using electricity to increase the number of electrons available to the bacteria's enzymes.
Can I answer this question?
Sure.
Yeah, that's about the correct answer, but not only the number, also the energy of the electrons. Basically, by providing additional potential, we are providing an external electrochemical driving force, which increases the energy of the electrons already delivered by bacteria through their enzymatic chains, and so the reaction is faster and results in the production of smaller nanoparticles.
And there's a second question from A Bit of the Universe. How do you control the electricity on such small scales? Is this on the scale of transistors in a modern CPU? Is the biofilm coated on something larger?
Well, that's an interesting question. At this point, we cannot control the electricity distribution, the potential distribution on a microscopic scale. Our experiment work with an average system, which is an electrode few square centimeter wide, and we only apply potential to the whole surface, and we don't care so much about the potential distribution. This potential distribution of the electron definitely will alter the production of nanoparticle in the system. Ideally, we should have a flat microfluidic system where the potential is applied in a much more homogeneous way, and nanoparticles are produced from the entrance to the exit of this microfluidic cell. This is quite difficult to scale up, and it's also quite difficult to set up for the moment. So we start with this simplified system. Biofilm is actually naturally developed on the surface. We are using carbon felt and other material because of the large surface and because of the bioavailability characteristic. We can also use other more defined surface like silicon, gold, or other bulk material. However, in this case, the current observed in the nanoparticle production is so small that we cannot go into further characterization of nanoparticles through microscopy or a spectroscopic method.
Okay, just going through the chat.
I hope this answered this question.
Yeah, I think so. Do you guys, do you have any other questions?
Not at the moment.
No, I mean, my main questions were, I think Marty really touched on it earlier, was kind of looking at what the applications of this will be possibly down the line. I know in your video, Kitch, on this, you mentioned things like, sunblock and stuff, which I guess was kind of out there, but yeah, what applications you could see coming from this technology further down the line?
Well, this will probably be this is more of another option to produce these nanoparticles. But there could be some side spin-offs. Sorry, I just noticed Enrico has gone offline when I've actually become what's the phrase? Convalent, or when I actually can talk. Typical, isn't it? But there are some side projects that could come out of this. For example, Enrico brought up bioremediation. That is the basically the removal of pollutants from the environment. One such thing we can actually do is increase the metal reduction rate, we can increase the metal removal rate from environmental samples. For example, there are some soils that can be contaminated with metals such as uranium, chromium, and wastewater runoff from gold and silver mines. You can put that into, we can use this technology to increase the removal rate of those compounds. Not compounds, metals. Let's see, Enrico is just dropped out. It's a Singapore connection. That's the one thing they don't have good there, is a good Wi-Fi.
Just looking through the chat, Microblogganism has asked a question: where would you naturally find Schwannella?
Schwannella you would find actually you would find Schwannella in the ocean, in the sediments, in ocean sediments. It's a marine bacteria, so it's a bit of the universe, as actually point out. Schwannella lohica is actually one of the organisms I'm working with. Was actually found off the coast of Hawaii. Not too sure if you have to start the call back because Enrico said he's back, but I'll just finish this anyway before I start the call again. And in fact, I think Schwannella lohica, or some species of Schwannella, are actually found in the intestinal tract of marine life. I'm just going to restart the call just to get him back. And there we go. Hello?
Hello?
No. Yeah, sorry. Sorry guys, this is I can see.
Oh,
There we go.
am I back? Yes.
Yes, actually,
I don't know how long my Ulysses work exposition was heard, but it's okay I'm okay with that.
Okay. Did you hear the question about where Chanel are usually found?
No, but I can answer. Chanella is usually found in sediment, particularly sedimented interface between aerobic and anaerobic zones. There have been Chanella found in Antarctica, Mississippi, Missouri, South America, in Argentina, in various lake sediment. North America in Lake Oneida, the first they were found. Some of them are discovered in Southeast Asia and so on. I think about 40 locations where Chanella was found so far, including a deep sea vent and 5,000 meter sediment.
And just at this point, I just like to remind people that the link to donate to the experiment.com page for this brilliant, beautiful work is actually in the channel description. It's the first link. It'll be experiment.com project page. So if you can donate anything, or if you're willing and able to donate, please do. It's just in the channel description. So actually, I'm not sure if this is a question or not, but I had one here. Now get pumped, link to channel. There we go. I see this is from Trollsar. I see you're using it as something to build non-organic electrical films or particles. Those are you're sizing nanoparticles, metal nanoparticles, particularly silver, though we're hoping that this will be used as a model to build on other nanoparticles, or to synthesize new other nanoparticles such as gold, palladium, maybe even if we can get it to that stage, we could maybe even look at bimetallic nanoparticles. Definitely not during the course of my PhD, but maybe on the further down the line by another PhD who's maybe interested in this.
Can I say something about that?
Sure.
Probably this person, I'm not sure I heard the name correctly, refers to the early project for nanoparticle deposition on surface. That was done using viral cage back in the late 90s, and it met some interest from computer producers, chip producers, because nanoparticle production on a surface produce a coating which is appealing for large memory. However, they are not so small as virus. Their structure tend to be less reproducible than virus because we don't have a specific protein cage available like in virus. And in general, the research is still quite in its infancy. The advantage of bacteria over other more specific systems like virus is the ease of growth, very low cost of the substrate, and so on. We are definitely looking at something like a nanoparticle solution rather than nanoparticle on a surface. Nano coating of bacterial origin is still a fairly new research field, and I'm not sure how much micro research will impact it.
And guys, do you have any more questions about? I'm not too sure. It's the mechanism about about how this happened. Is that clear? I'm not too sure if that got actually through all the cuts and my general incoherentness.
Well, it's probably just that it's above my head. I guess that's the problem. But I actually do have a question, and it's gonna. I understand that to you this is gonna sound stupid, but when you produce nanoparticles, are we talking about taking like a chunk of metal and breaking it down into tiny pieces, or is it like finding more or less loose atoms in a solution and putting them together into nanoparticles, or how does this work?
Oh, okay. That's actually an interesting question. What we actually are doing is that we are exposing the biofilm to a salt. So basically, it's metal ions just in solution. So we're just basically building it atom by atom,
Yeah.
which is actually in a process known as bottom up. It's a bottom up process. You can go the opposite way and go from the top down, and as in take the metal and break it apart using high energy such as lasers. But that kind of the disadvantage about using that is that you can leave imperfections in the nanoparticle surface, which affects the nanoparticle's activity, and that's not really desirable. So that's why bottom up is more preferred to the top down. Although, as far as I know, top down people are still working on top down, but don't quote me on that. I'm not actually that far into the nanoparticle side of things. I'm still more biotech.
Okay, let's say something on this, Michael. I don't think there is actually enough energy in the bacterial reaction to bring down metals as the laser do. What has been done in the past has been the oxidation of metal from an insoluble to a soluble state. For example, the famous experiment with manganese done by Ken Nilsson in the early 2000s in his lab in California, where bacteria are able to eat a chunk of solid manganese, which is called manganese
Oxide
4. Yeah, manganese oxide 4, and reducing to soluble manganese. So that's actually one of the first evidence of bacterial reduction in the environment. However, the reduction of the destruction of this material doesn't result into nanoparticle, but into ions. So the other process is possible when we take ions and aggregate into nanoparticle together with the aid, and that's a project of Michael, with the aid of electrochemical potential. As you probably know, in the nanoparticle formation, there are two stages. One is the seed part, the seed process, and the other is the growth process. In the seed process, few atoms come together and remain together as nanoparticle, due basically to sort of random aggregation of atoms in their insoluble form. After that, the small nanoparticle, few nanometer, grows with time, particularly in presence of capping agents that prevent aggregation of this nanoparticle with other particle. So what we are working on is accelerating the rate of seeding to obtain smaller nanoparticle, so that the subsequent growth phase and capping phase, where the capping agents of biological origin limit the size of nanoparticle, result in a focused small nanoparticle distribution. I hope that this answers your question.
If I could just maybe even just reword it just a little bit, we're hoping to, by increasing the metal reduction rate, to create basically smaller, a greater number of nanoparticles, but smaller number of nanoparticles, which should bring down the size distribution, or in other words, increase the uniformity of the nanoparticles, so we get nanoparticles that don't really differentiate from each other in size. So we can, did I get you kind of summarize that quite correctly, or?
Yes. Does it answer your question?
I suppose.
Okay, fair enough.
So I just going to. Is there any other questions? Actually, I've an interesting one. You'd mentioned there, Enrico, bacterial oxidation.
Yes.
Is there? I actually brought up in a blog post about bacterial corrosion. Can you actually use electrochemistry to study how bacteria would corrode pipes, such as mortar?
That's one of the oldest application of bioelectrochemistry, that dates back to the 80s or even before. And in Singapore, we are now building a new research line in biocorrosion from native marine community. There have been several projects on biocorrosion in the past. Basically, what's happened is that bacteria, as a single cell, floating cells or biofilm, either protect the material from oxidation like a typical passivation layer, or in certain case, increase the availability of sulfate locally, by so resulting in a faster oxidation rate. This effect is quite complicated, and it's not clear when the biofilm favor oxidation or prevent oxidation. However, there is an effect particularly strong in salt water. Electrochemistry is a typical method, so far is the best method for this investigation, to get across because the rising of change in local impedance and change into the slope of the Tafel plot, which is basically a polarization plot, and these slopes give an idea how much corrosion is increasing with time. So yes, corrosion is quite important for our research. Michael is just Michael project is only tangentially related to corrosion, but in Singapore, when I moved a few days ago, we have a very strong research line on that.
Sorry, I'm just going through the chat again. It won't keep up with me.
Just had a question here from Bit of the Universe.
Okay, just got it
how
there.
are the nanoparticles extracted and stored?
Oh, okay. Can I take this one?
Please, Michael, go.
Sure. Okay. Right. When we produce, after the experiment is finished and we have produced the nanoparticles, we have basically now a suspension of bacterial cells, nanoparticles, and what the bacteria has spit out. Basically, Trinella spits out or secretes this compounds known as flavins, which are involved in its electron transfer mechanism. Basically, its ability to reduce or transfer electrons to external electron acceptors, such as the electrode or metal ions. So basically, we need to separate the nanoparticles away from all that other stuff. So what we basically use a very simple method. It's centrifugation. So basically, we can pellet down the bacteria, the bigger bacteria and the bigger bacterial debris from the nanoparticles by a simple centrifugation, a relatively slow centrifugation for about 10 minutes. That will leave us with just the nanoparticles and the secreted compounds, these flavins. We then take this, we take the supernatant or the media, the liquid that is left over. When you centrifuge, you'll have a pellet and a liquid top. We take it and we centrifuge it again, this time at a higher centrifugation, at around 12,000 times the gravity. 12,000 times gravity, so basically it's 12,000. These nanoparticles become 12,000 times heavier. So they will collect down at the bottom of our tube. We can then remove the liquid, because the nanoparticles are now collected at the bottom of the centrifuge tube, and then we resuspend in distilled water. And basically we store at, I usually be cautious, I store at 4 degrees in the dark, because the silver is quite sensitive to light. So we need to store this under tin foil and at 4 degrees, just to be absolutely sure, because we're not too sure on the stability of these nanoparticles. This is something I'm actually in the middle of doing. I'm actually just waiting for a few more weeks for these for the nanoparticles I produced before I check them again to see if they are corroded.
Michael, I have a question.
Yes.
All this what you are saying is just fine for laboratory practice, but maybe the Bit of a Universe was interested in asking, is there any technique for large-scale separation of nanoparticles? You think that you know something about that?
As far as I'm aware of, that's actually something I've been trying to find out, but as far as I'm aware of centrifugation, it would be still to probably be the there may be other filtration methods. Actually,
well, let's say that part of Michael's PhD will be also to determine if we can design a new reactor where we can immobilize a newly synthesized nanoparticle and possibly wash them with a single wash step, and we can retrieve them at high concentration. The problem is it's not so much the problem to separate nanoparticles from cellular debris, because the size is very difficult and centrifugation will work. The problem is how to separate eventually a nanoparticle of certain size from other sides without removing the capping agent that prevent aggregation of nanoparticle itself.
Mm-hmm.
Okay?
Yep. And sorry, I just got a bit distracted there. Have you got any more questions?
There was another question in chat from Trollzar asking, could you potentially use a two-stage process using the bacteria to generate the nanoparticles until it reaches its size cap, then use a secondary electrical process to build upon the existing nanoparticles?
Okay. Michael, are you going to answer, or I try to answer?
Okay, sorry, I just want to go back to the original. There we go. For the bacteria generating a secondary electrical process to build upon the existing nanoparticles. I'm just trying to kind of process that. Well, at the moment, we're actually considering using to generate these nanoparticles until it reaches the size, until it's finished, and then reduce the size further using an electrical, using an oxidative potential. But Enrico, I think you can answer this one better than I can on that one.
The question is interesting, but there is a problem with that. Let's try to explain. We don't want to produce large nanoparticle. That's fairly simple and can be done with any kind of reducing agent, from geranium leaves to reducing sugar to graphene. Anything can be a reducing agent. We want to produce small nanoparticles of a focused set distribution. So the electrical potential is not applied to nanoparticle large gates or nanoparticles, but to nanoparticles application. Maybe the listener has in mind another application when you produce they're called sort of structurally long distance order nanomaterials through consistent application of electrical potential. That's an interesting field for nanomaterial production, and is used, for example, in computer science, in silicon technology to produce whiskers and other things. However, our goal is to produce a variety, produce a high concentration of small nanoparticles which are good for biological application, not large nanoparticles. It's still interesting to combine electrical and biological steps, for example, in a cycle way, and that's definitely a suggestion that we take in.
Okay. And actually, I just want to kind of get through the questions because we're almost finished with our hour. Ability Universe asks, I'm curious about these Shunella and their ability to be both aerobic and anaerobic. Is this fairly unique among bacteria? Seems like a pretty significant adaptation. And well, the ability to be facultative anaerobic it's actually not unique to Shunella. There's another genus, Geobacter, that's actually been investigated for their applications in bioelectrochemistry, but as preferred for our purposes because it is a strict anaerobe, no oxygen allowed. Yes.
I'm not sure the listener want to say to ask something else. is it normal that bacteria can operate both in aerobic and in anaerobic conditions? So how many bacteria in the world basically are facultative? Well, many bacteria are facultative. However, few of them are really facultative. Facultative bacteria are so bacteria can respire under a non-aerobic condition using fermentation, where the electrons for the respiration are disproportionate between a red and an ox compound. Okay, that's a typical respiration for E. coli that produce lactic acid as a final electron acceptor, and yeast that produce ethanol as electron sink. So that's a common mechanism. However, it's much less common to find bacteria that are able to interact with solid surface and are also facultative. Shionella is one of the few that we know. The other one, some the sulfovibrio can be also called facultative and can work under this condition, but Shionella is pretty much the most investigated bacteria, facultative bioelectrochemical bacteria that we know.
And is Pseudomonas, is that another true facultative anaerobe? Because I know that one's actually used in for electrochemical purposes.
Yeah, but Pseudomonas is not capable of reducing directly a solid electron acceptor. It can do this only through stimulation of production of mediator like phenazines. However, phenazines are produced only under aerobic conditions. We have a couple of papers published in Singapore about that. So Pseudomonas is not really an electroactive bacteria, although it produces a radioactive compound that can result into energy production.
Okay, and this is actually, I think this is just more of a bit of a joke. Actually, and this is just a bit of a joke, but I'll kind of end on this. A bit of the universe says sounds like making a multi-layered jawbreaker with a gold core, silver mantle, and copper crust. But anyway, that's all our time for this hour. We staying tuned for Phil Moriarty, who's coming up next to talk about science funding. I'd just like to thank my supervisor, who kindly agreed to join us at this this late hour, being over in Singapore. Stay tuned, guys, and don't forget to donate. And if you have any questions, please feel free to contact me through the experiment.com page, or you'll be able to contact me through the Trolling Limit Logic website. Thanks, guys.
Okay, thank you, Michael, for hosting this. See you next time.
See you next time.
Yeah.
Thank you to the others. Bye-bye.
Bye.
Cheers.
Cheers.
Hello.
Hello.
Hello?
Yep, sorry. Just going to make sure everyone is around.
Hello.
Hello. I'm doing the tech today, so expect to be quite buggy.
I'm sure it won't, Michael.
Well,
Can you hear me okay?
yeah, I can hear you all okay. We're just live right now because I'm just going straight through with the 3 hours, so it just makes it easier to record and download, because I'm expecting this computer to bug out at multiple occasions.
And you're doing it from the lab as well.
Yeah, I was actually just came straight from a meeting, so the first hour I was brain fried. Okay, so thanks for coming along. Welcome to hour 3. Just at this point, I just want to remind everyone again to there's the link to contribute to the experiment.com portal. The experiment.com project is actually in the channel description, and it should be right below. It should be the first link. Please, if you're willing and able to contribute, please do so. So, I think we agreed for the topic to be about science funding.
Yep.
Now, with the economic recession at the moment, it's quite hard to get funding here in Ireland. Is that the same? I would assume that'd be the same over in the UK.
Yeah, it's much the same. It's always difficult to get funding. I guess at the moment, I don't know exactly the success rate, but I'd imagine it's somewhere between something off the order of 20%, 20 to 25%, something like that. And I know in Ireland, I think times are certainly very tough now, particularly for fundamental research.
Mm. And over in the UK, there are how many? I know for the Ireland here, I know of the SFI grants and the Irish Research Council grants. The UK being a bigger country, would it have more grants available?
So there are the major funding of research. It's very interesting and very complicated over here. We have something called a dual support system, which I'll get to in a second. It can get very tedious and very boring very quickly. But the major way of getting funded, certainly for a physicist, is through one of the research councils. So there are 7 research councils, 2 of those being, for example, the Engineering and Physical Sciences Research Council. There's also a Biological Sciences Research Council and a Science and Technology Facilities Council, as well as the Arts and Humanities, etc. So they have the bulk of the funding. They supply the bulk of the funding in terms of research grants. Then you also have charities, for example, like the Leverhulme Trust. You also have the Royal Society, for example, Wellcome Trust. There are a range of different funders, but in physics at least, which is my area, the bulk of the funding comes from the EPSRC, the Engineering and Physical Sciences Research Council.
So I just got to work the tech just a little bit. And that's sorry, just a little bit stubborn.
Well, just very quickly, Kitch.
Yeah.
I noticed on Vaughan we can't see.
I know that's what I was. That's what I was just kind of correcting for there. Just for some reason.
As cute as my dog is, I think we're more interested in him.
Just fixing
Don't know about
that.
that. Your dog's fairly cute.
And apart from the UK government grants, there are also European-wide grants that you can apply for as well, such as the Europa, if I got that right.
So Europa I haven't applied for, but one thing I have for, there are a wide range of what are called Framework 7 and now Horizon 2020 grants. This is multi-billion euros across the board, and they fund right across Europe. So Nottingham at the moment coordinates what's called a Marie Curie training grant, which funds PhD students. And the one I'm involved with funds, 14 different students in 6 different countries. The Marie Curie, in terms of the funding, maybe I should send you the proposal sometime, Michael. It's I can't remember. It's off order 100 pages long. It is very specifically, an awful lot of it is to do with management, and an awful lot of it to do with this process rather than the actual science. The science is like a 2 or 3% perturbation on top of the overall management case. And Marie Curie, I would say, is about the best. There are other schemes we're involved in, other projects where it's shocking in terms of you have to lay down the deliverables right at the start of the project, and then you have to deliver those deliverables. Even if the science doesn't seem to be going anywhere, you get locked into delivering deliverables. And it's very bureaucratic. It's a very short-sighted way of doing science. On the other hand, there is something called the European Research Council, which does it right. The European Research Council focuses on the science and focuses overall on the quality of the science. The problem with the European Research Council is the success rate is something like, 3 to 5%. So it's very tricky to get funded. And certainly in nanoscience, when you look at the people who are funded for ERCs, they look to be in the cusp of a Nobel Prize, basically, in many cases. So for us mere mortals, it's tricky to get in there.
So I guess, Phil, would that mean that trying to do blue sky kind of science is fairly difficult in the UK?
Oh, right. I could spend, as Michael probably knows, the best part of 4 hours discussing this. In the UK, certainly I'll stick with the Engineering and Physical Sciences Research Council, which is the one I know the best. We now have to, with every grant application, you have to fill in not only a pathways to impact statement, but you also have to fill in a national importance statement, where for pathways to impact, what they argue, what the research councils argue you do, is that you should identify the users of your research or the beneficiaries of your research in advance of doing the research, and then indeed to those beneficiaries. That's pretty far away from Blue Sky's research, and in fact, in the worst case, you could argue it's actually counter to the scientific method, because if you're doing research to reach a particular target, quite whether that's fundamental or basic science is a very, very moot point. It's closer to, you know, commercial R&D. Yeah. Which is very troublesome. It's something that's irritated me and many others quite a bit over the last number of years.
Yeah, it seems like that sort of environment isn't exactly going to make people find the next Higgs boson type thing.
Absolutely not. No, it forces you to think very incrementally. It forces you to think, you know, what industry is going to be involved with this, what industry is going to be interested in this, can I push this so I can get some commercialization out of it, what's my intellectual property, all those type of things, you know. I'm not arguing that there isn't a place for that. Of course there's a place for that, and DCU is very good, for example. Dublin City University, which is my alma mater, it's where I did my degree, is very good at connecting with industry, as are many other universities, including Nottingham. But that, for me, is not the fundamental role of a university researcher. The fundamental role of a university researcher is not to act like an outsourced, you know, as part of the outsourced R&D wing of a corporation. It's to do work that's, particularly if it's publicly funded, that is independent and pushes the boundaries. And indeed, you talk to many people in industry, and that's exactly what they say. They said they don't want universities to be doing exactly what industry is doing, that there has to be, because they, you know, often they don't have the funding, they don't have the money, particularly in these straitened times, to pursue those type of disruptive technologies, if we want to put it that way, this type of disruptive research. As you can hear, I can get fairly exercised.
It's something that bothered me as well when I was picking my career and decided on becoming a teacher instead of a physicist. The whole idea that being a physicist seemed like, I mean, yeah, I would be fascinated to you know, get paid to ponder the great mysteries of the universe and do fancy math about black holes and stuff, and that seems really fascinating. But then someone comes along and says, yeah, but who's going to pay you to do that?
Yeah,
they pay you to develop this screw for this engine.
Yeah, I agree, John, and indeed in my darkest hours I very much thought about jacking it all in and going off to do teaching myself, actually. But you can still, it's tough, but you can still get fundamental research funded. About 6 years ago, I got very pissed off, along with a number of others, about this whole drive towards impact, and particularly the idea that you should let the impact define the direction of your research program, that you should let your predicted beneficiaries, or the people you think will be the users, define how you do science, and that's really uncomfortable. So I actually decided at that point, I'm not going to do any grants. Well, I'll review grants, but I won't review that bit. But the Research Council said, no, you can't do that. So I stopped reviewing grants, therefore I stopped submitting grants. And it's only actually in the last few months that I've gone back and I'm about to actually just click the button before I click the button on Skype here to talk to you. Submitted a proposal for the first one in 6 years to the EPSRC, the Engineering and Physical Sciences Research Council, on the premise that they argue, and I'm going to do the experiment. They argue that no, we are just as interested as we ever were in fundamental research, and your proposal won't be disadvantaged if you are doing fundamental research. So it'll be interesting to see how this one is received. My pathways to impact, as they put it, and my national importance are fundamentally and solely about public engagement, because that's, if you're publicly funded, you have an obligation, you have a duty to communicate what you're doing to the public. So it's biased that way rather than the commercial side, and it'll be interesting to see how the reviewers respond to that.
Yeah, and actually, there's always there was actually a joke. Actually, I've actually quite heard that if you know that if an editor gets reviewer 3 to be come back with positive but a positive feedback, you know that it's sorry, my mind is just gone from today. First day back in work. When reviewer 3 comes back with a glowing glowing review, then you should start to suspect fraud.
Yeah, it's interesting. It's generally peer review's flawed. It's, there's the old cliche about democracy being the best thing we've got, and peer review is absolutely flawed. We've been, this striped nanoparticle stuff, Michael, we've been embroiled in that over the last couple of years, and absolutely that really brought it home to me, how flawed peer review can be. And if you some researchers, number of groups have taken the same set of proposals or the same set of papers and given them to different peer review committees and got dramatically different results. So it's flawed. It's always flawed, and as you say, you can end up with 3 reviewers, 2 of which will think it's the best thing since sliced bread, and 1 will think it's the biggest pile of crap they've ever seen, and vice versa. So it's tricky. It's very tricky.
And is that the same with?
Why your experiment with this experiment? Sorry, what I was about to say, Michael, is why your experiment, this experiment.com is very, very interesting. I'm extremely interested, almost fascinated with the sort of crowdfunding aspects of this. It's a really, how can I put it, thought-provoking way. It's a really, just to see how this pans out. And I'm not saying that we can obviously fund all science like this, but in terms of crowdfunding schemes like this, it's clear that they have a role to play.
I know that's I've started this project as out of interest. I am actually approaching the final leg of my PhD. Hopefully, if I haven't just shot myself in the foot with that. And I'm just hoping to receive just the last bit of money, just so I can get the last bit of visible and the last bit of money for the metal. As I'm working with heavy metals such as silver and gold, which as you know, are very, very expensive.
Yes, indeed. Yes, it's so I'll be very interested to see how this pans out, because certainly most of the crowdfunding stuff I've seen has been very much targeted on a specific product or deliverable. Although fundamental research has got funded, it's it's a much harder sell, much harder sell.
Yeah, and we actually had a research. We actually applied for SFI funding for a postdoc. Actually, a former PhD student of ours who's actually just gone off actually now on a Marie Curie grant to Italy, and as far as I remember, we got the grant application back, and it was just not enough experiments to do. They said that the background work wasn't really there. And at one stage, actually, one reviewer actually contradicted himself, and I don't want to kind of go into too much detail about it because I don't want to get anybody in trouble. I'm not too sure about what I can't say about these kind of grants. But it was going through it. I remember just being quite frustrated with the way the grant works. But sure, that's probably more disappointment than anything, really. Kind of things. But as far as I know, we actually did get a grant approved based on the project we're actually working on now, but for another PhD student who'll be coming in after I leave with Europa.
How long have you got left, Michael? Did you say?
6 months.
6 months. Okay.
Probably will go over, but I've got money saved in the bank just to keep myself going that way. It's just the bacterial cell work that I'm actually working on now is just in the last legs, but I'm actually working with mammalian cells at the same time, and though that's that they take a lot longer because they're more complicated, and we're actually developing a mechanism about how the mammalian cells actually produce gold nanoparticles. This is actually, and then we actually go into a lot more detailed analysis, looking at genetic expressions and stuff like that. And that actually is quite expensive. Luckily, that part of the project is being co-funded by another supervisor, but he's only looking after that side of the project.
Okay.
Because that side of the project is well outside anything we could probably raise on experiment.com. You could, for a mammalian cell project, you need about €10,000 per year, and that just might about cover the cost of media, reagents, use of all that sort of stuff. And then if you have contamination.
Yeah, it's interesting. I don't think it's widely realised, but for those outside the system, and there's no reason why it should be, just how much it costs. So the grant I'm about to put in, which is basically just to fund a postdoc for 3.5 years, some travel, some consumables, not a huge amount. When you build in the UK, what are called estate costs and indirect costs, effectively overheads associated with that, it comes to, for a 3.5-year grant with one postdoc on it, no equipment, small amount of travel, damn near $500,000. So it is quite remarkable how quickly those costs, tot up. And then when you think about, the total budget across all research councils is $3 billion per year. That seems like a huge amount until you work out that if a relatively small grant is $500,000, it doesn't take so many of them to get to that point very quickly, and hence competition is intense.
Yep, and that's all. As I say, it's all facing me in a 6 months' time, well, hopefully. So that's why I thought it's also another reason why I went for the experiment.com, a bit of experience.
Yeah. And you're interested in pursuing a postdoc and then onto an academic career, or?
Actually, funny enough, I did get, when I was actually in ESOF last year, or 2014, and I went to a career advice booth, and they actually kind of took me through almost a step ladder. I actually have it up in the office, a timescale. First postdoc, 2015, fingers crossed.
Scary, isn't it? Yeah.
Yeah, and then a second postdoc, and then actually they advised me to apply under Horizon 2020, which I actually have kind of, that's a bit far away. But actually, this actually might be a next question. What actually is Horizon 2020? Because I've heard that an awful lot throughout maybe conference, when I was arranging for speakers as well for the Biological Research Society. I heard that come up as well for meetings, had to rearrange kind of talks around those 2020 meetings.
Themes, yeah. Yeah, did you say what is a Horizon 2020?
Yes, what actually is Horizon 2020?
So the European Commission has, in previous rounds, has had what are called its framework programs. So these are huge, basically billions upon billions of funding right across Europe. And it's has Framework Program 5, Framework Program 6. We're sort of in the end part of Framework Program 7. So instead of having Framework Program 8, they have Horizon 2020. So it's basically just a name for a whole set of different European those platforms are very targeted, very particularly, look in nanoscience alone and nanotechnologies, you'll find literally hundreds of themes, and then very specific targets, very specific calls. Marie Curie again is one of the few that allows some degree of a large degree of flexibility compared to others. So it's really just a catch-all term for a large number of European Commission funding programs.
And I imagine competition for that is going to be quite unreal.
Yeah, I don't know what. So the one I target, I wouldn't. Yeah, I suppose it's fair to say I target. The one I go for is the Marie Curie side of things. A, because there isn't such an intense and stupid focus on research deliverables, and B, because the level of reporting is much less onerous than it is for other schemes. So for Marie Curie, I think the last round for the networks, the training networks, that was something like an 8 or a 10% success rate. So it's, yeah, this competition again is very intense.
And actually, Nottingham actually, I understand, has a higher than average.
Yeah, Nottingham does, and one of the reasons for that is we employed for a while, for a long time, a consultant who actually, you'd send your proposal to him, and he would read over, give you comments, etc. So there was a fairly heavy filtering and sifting before it went ahead. So there's that. There's the fact that also UK UK applicants tend to do on average a little bit better with Marie Curie than others. Whether that's a language thing, I don't know. Nottingham in the UK does pretty well in terms of Marie Curie training networks, at least.
And Marie Curie is not just one grant, but there's a selection of different grants under Marie Curie.
You can have individual fellowships. We've got a couple of people here now in Nottingham are funded by individual Marie Curie fellowships. So these are 2 years long. And they're really good because they allow a postdoc independence. The postdoc can basically write, or not basically can, is expected to write the proposal. It's meant to be coming from them. It's meant to be their own independent ideas. So it's a good way of getting your foot on the funding ladder and supporting yourself for a couple of years. So it's good schemes. Leaving aside the usual qualms and usual moans we have about any type of funding scheme, Marie Curie's not that onerous.
And actually, I just want to take this point of time to remind people that they can still contribute to the experiment.com project. The link for that is down below. If you're able and willing, please do. So actually, Marty and Ziller has been awfully quiet over the last few minutes. I just don't want to hog all the questions. You guys okay? You guys have any questions?
Not really.
Is there? Sorry, I've been trying to keep up with the chat, but it's kind of gone a bit. Is there any questions in chat? Oh, there's one from a guest. Do you find you have to sell your idea, display how to make money from your idea, or are ideas funded based on other aspects?
That's a really good question. So the advice under some of the criteria for the grant applications, again, I'll stick with EPSRC, is one of the things they specifically say is how might this work bolster the economic competitiveness of the UK, for example, or contribute to the GDP of the UK, whatever. However, you don't have to go down that route. At least that's what they say. This is it'll be interesting. I'm just submitting a grant, as I say, which basically forgoes any mention of commercialization. So that is, is that going to help? I think if you do mention that, yes, this could lead to this commercial spin-off, it might impact on this particular sector, et cetera, et cetera, et cetera. That's the type of thing that EPSRC, the funding body, really likes to hear. The question is whether, it's necessary for other academics who are, after all, the people reviewing your grant, to convince them, do you really need to mention the commercialization, or indeed is the commercialization actually going to adversely affect the chances? Because one thing that's a real problem is that you can write 2 pages, you're expected to write 2 pages in terms of the impact. Any academic, I think, worth their salt can churn out 2 pages of purple prose, fictional purple prose, on how their particular research is going to change the world. Demonstrating, having definitive evidence or being able to point to, hard numbers or hard evidence that actually you are going to have a commercial impact is another matter entirely. So I just think the idea of, arguing that we should have a pathways to impact case, we should be trying to predict the commercial impact of our work at the proposal stage. It's just stupid. It's just really, really stupid, because you end up writing, science fiction, basically. You end up writing science fiction. Whether that's entirely necessary, is another matter. You know, it is clear that very good fundamental research is still getting funded in the UK. The bias, the skew, is definitely heading towards more applied research, but fundamental research is still getting funded. I have huge problems with the system, but you know, it would be disingenuous for me to suggest that it's impossible to get fundamental research funded. So, not as depressing a picture as I might have painted earlier on.
I know Enrico said when he was in Ireland that if you wanted to get a research grant funded in Ireland, you have mentioned cancer in it somewhere.
Oh yeah, of course, yeah. Yeah, it's unfortunate, but that's you know, the buzzword. You know, nano, to be fair, is another buzzword. Not so much now, but certainly 10 years ago, you know, if you could get nano shoehorned into the title of your grant, that was seen as a good thing. You know, you can point to graphene now. You know, graphene's going to save the world. So
there was a molecule. It was not molecule, but it was material of the year 2 years ago, 2012.
Probably, and that's not. You know, graphene has got some really, really wonderful things about it. Its electronic properties are very different, very distinct, unique in the proper sense, and really fascinating. But is it going to, you know, revolutionize everything? Is it going to be a panacea that cures every single societal ill? I don't think so. And sometimes that's how it's sold.
As a magic material, almost. That's what I kind of got from it, from some reporting of it. That was just, if you got any ailment, just put some graphene in it. It's like that quote out of what was it? My Big Fat Greek Wedding, the window lean.
Yeah, it's very, very heavily hyped, and then there's a big bandwagon effect on that, and lots of people jump on the bandwagon, and it's sometimes a little bit dispiriting to see, you know, that bandwagon effect in action, where so many go, oh, there's money there, let's chase the money.
In the UK, is there any particular research areas that are more funded or preferred to be funded?
Yeah, so this is another big bone of contention. There's something called shaping capability, which was introduced a few years back, whereby many areas, including surface science, which is pretty well my research area, was targeted for a reduction in funds. So you have different areas labelled expand funding, maintain, or reduce. So there are very much preferred areas, and many of those are increasingly driven by what the research councils think the government wants to hear. Because it's a very interesting, very complicated, very boring dynamic between the research councils and government. Because research councils are not a government department. It's interesting, they're meant to be at arm's length from government. There's something called the Haldane Principle. There's a huge history behind this, which I won't bore you with. But the idea is that the research councils should be, as I said, at arm's length from government. They shouldn't be jumping to, you know, the government's will, and yet that's what's clearly happening. They're thinking about how best can we present ourselves to the government so that we can get as much money as possible in the next comprehensive spending review, in the next budget. And you know, in one sense, that's laudable. They're trying to get as much money for science as possible, but the question is, you know, what are you giving up to get that money? What are you selling, you know, in terms of integrity, credibility, and supporting fundamental science? So there's a huge debate about this going on at the moment, of course, because we've got an election coming up this year. It's going to be an even more intense debate coming up to that election. I'm afraid, Michael, I'm going to have to, which I hope I told you in the email, I'm afraid I'm going to have to go in about 5 minutes' time.
Sure, no worries. I'll be able to fill in. I'll be able to fill in the. Fill in. Sorry. Yeah. No worries. Yeah, I got the email.
Okay.
So yeah, I've got something to fill in that. But sure. Guys, do you have any questions? Because I feel like I'm just kind of keeping this, you know, being not taking more than my fair share of the time.
I know, I always have lots of questions in my head when Phil Moriarty's on, then suddenly I get the chance to ask them, and they all just disappear. I'm gonna have to write them down one day, so I've got them all in front of me.
Yeah, then there's a chance one might go completely off topic when one has a physicist to play with. We had our Ziller, we had our little problem that you brought up to me that with the light thing. Weren't you arguing with flat earthers or something, and that's where it came from? Like how, if the Earth were just a flat plane, how far would you be able to see?
Yeah, at what point would the atmosphere appear, I guess, for want of a better term, opaque? At what point does atmospheric extinction extinguish all visible light? At what distance that happens?
You know what? I don't know. It's a very, very interesting question. Off the top of my head, I really don't know. Yeah, light is a really strange thing, and light's interaction with matter. You know, that's what Feynman won his Nobel Prize for. So it's a pretty tricky concept, and lots of even looking through a piece of glass. So much physics going on there in terms of something that's apparently transparent, but then what's happening at the surface in terms of the reflection. What's happening in terms of refraction? It looks like the light goes through unimpeded, but of course it's not unimpeded because otherwise you wouldn't have refraction. What's happening at the quantum level? What's happening at the wave level? It's really fascinating aspects of that. But to answer that question, I'll go away and have a think about that. But in terms of the absorption and the scattering,
exactly, it didn't sound like it should be that much of a problem.
Well, it's interesting.
We all kind of know what goes in there, but how do you actually calculate?
Yeah, well, you'd have to think. Right. So first of all, there's a range of different. So with any optical problem, you're going to have a wavelength dependence in terms of the absorption and the scattering. So you've got to think about what that wavelength dependence is, because that will change your effective extinction coefficient, whatever you want to call it, depending on wavelength. Then you've got to drill down. Is it actually absorption or is it scattering? Then is it scattering due to sort of Mies scattering or Rayleigh scattering, or is there some type of Raman process occurring? And if it's absorption, what type of absorption is that is? And then readmission in terms of you might excite an atom or a molecule to a particular excited state. How long does it take for that state to decay? What sort of wavelengths are you generating in that process? Huge numbers, which generally what you do as a physicist is trying to bring these all together, all this physics together into one simple number, which is like a decay constant. But the physics behind those decay constants are really quite complicated. And I'm afraid on that particular note, I'm gonna have to leave it because my wife's working tonight, so I really need to get home and start looking after the kids. It's been a pleasure as ever.
Grant, sorry, just have one more.
See you then. Bye.
Bye. Oh. But I just have a question for us. I don't think. Sorry. I have a question from Trozar anyway. So have you guys heard of gridcoin.us? It's a cryptocurrency similar to Bitcoin. I haven't heard of it. I think it's a bit obscure. Sorry, I couldn't ask Phil, but no, that's just live broadcasting. So anyway, I'd like to take at this point to remind people they can still donate or contribute to the experiment.com page. It's linked in description. We do have a donation from. Actually, I'm just going to check my email because I've lost it. Sorry. From Microblogganism. Thank you for your donation. So how can we fill in between now and actually I'll take questions about the project. Maybe there's something that wasn't really explained well that can happen because, as I said, I did come into this a little bit brain fried from a lab meeting that went on far too long than I had to for the first day back in work. So, is there any questions about what the project is about? What's the science behind it? What's the application? What's the mechanism of how this is happening? The bacteria, the nanoparticles guessed, will it blend? Possibly. If you're willing to donate money, I will answer that question. I will do the experiment to prove that question. There's your money at work. I'll find out if the nanoparticles will blend. So I wish I even had. I wish this didn't occur on the first day, but I didn't want to leave it too late. Because I'd have an electrochemical cell up and running and be able to show that off. I do actually have the bacteria up and running. Actually, I came in yesterday. I'll be back in a second.
I was hoping Hitch might give us a tour of his lab at some point.
I would, but literally I would need to unplug the laptop, and the battery doesn't work in this laptop anymore, so as soon as I plug it out, it dies.
Damn.
But actually, what you can see here is actually my bench space, this actual lab bench, although there's no point in saying it's my lab bench anymore, because I'm the only person left in this lab for the moment. Yep, I'm here on my own in this lab. But
what does that say about you, Kitch?
That I'm just not a nice person. But actually, as you can see here, I actually came in yesterday because I have no life, and I took a bacterial cell up. Basically, when we store bacteria, we need to. Oh yeah, I need to put my window on larger. How do I do that? How do I actually make myself larger? Nobody. Did that work? Not too sure.
I want to do a study on this because every time I've seen one of these live hangouts or something like this on Vaughan TV, it seems the more scientifically minded the person doing it is, the less technologically able they are.
I'll take that as a compliment. Well, anyway, yeah. When we store bacteria, basically you can't have bacteria out all the time because they'll accumulate so many genetic changes and you won't be able to use it anymore. So basically we store them in stocks at a temperature of -80. That means that the bacteria just doesn't grow and doesn't metabolize. And you also add in a bit of glycerol, so that basically prevents crystals from forming, ice crystals from forming. And that can damage the cell if it's formed in the cell because it'll start bursting the membrane and leaking out all the bacterial guts all over the place. So this is actually what I have in my hand, a culture of Shigella. Now I took this out yesterday, so the culture isn't that active at the moment. It needs a little bit of time before it can actually get back into the full swing of things because -80 is quite harsh, so it's going to take a little bit of a while for the bacteria to grow. But what I hope you can see there are bacterial colonies on this plate, I assume.
Yeah.
And basically this is
I hope that's what it is, because I think you can get banned for...
Yep, no, they're not lines of an illicit substance. Not yet. And basically, you can see here that the color is orange. Basically, why that this colony is orange is that on this LB it's known as an LB agar plate, but I've added in iron, about 1 gram per liter of iron, and the bacteria basically produce iron oxide, and that gives you the orange color, or the iron oxide is rust. And also, yeah, it smells, because bacteria they're smelly and they don't like to be added to -8 well they can usually do well. But I'm going to put these back anyway, just so I don't forget about them. Be back in a second again. Oh, and yeah, and since my supervisors are not looking, I do have these. That's actually gone a bit off colour. That's actually okay. I do have bottles of wine in the lab, basically from the last PhD moving. Had a bit of a celebration, and the lab mascot, Barry the sheep.
I was going to say the wine's not part of an experiment then, Kitch.
Maybe at the end. You can probably guess the experiment. And oh, actually, I do actually have samples of gold here. Actually, see, these are actually on a related project. I basically take the surface proteins from a fungi known as rhizopus, isolate them and incubate with the gold salts for about 48 hours, and then the fungal proteins produce these lovely nanoparticles and different types of ways that actually what we found that the way that you isolate the proteins from the fungi actually influences the nanoparticles produced. I hope this isn't turning out into just absolutely dry, plain, boring filler between now and when Myles finally arrives. Actually, hold on. I think I know how to. Actually, oh, I made a serious error or flaw here. I will. Now my Mimi.
Oh, hello.
Oh no, we've got him.
Fear me and obey.
It won't be a Trollum logic without Angry Pumpkin.
Of course, I'm the nicest, kindest, most polite person in the universe. And now that I've stopped lying like a politician on crack.
Happy New Year, you bastard.
Please don't remind me that it's the new year.
A new year.
Makes me think that I missed out on opportunities to get drunk last year.
Or maybe opportunities to get drunk this year.
I like your optimism. Now, please tell me what you're doing, because I have no idea.
Well, Phil had to leave a little earlier, and Myles couldn't actually join us until 6 o'clock, so we're just filler.
We have a quick question from guest 264 who wants to know if Marty can see his chakras.
And I will dignify that with a response.
Assume that means yes.
Well, I will answer the question. Guest 264, I can't see your chakra, but I can see your penis. Please put it away. And just wait for the awkward silence to fall.
I have literally no idea how to respond to that.
No one ever does.
So, any other questions from the chat while we are waiting for... Oh, actually, Myles is online, so I'll be back in just a quick sec.
I have a question.
Shoot.
How do you science?
With a spoon.
Good answer.
A wooden one.
The constant heavy breathing from Kitch is making me think I'm involved in phone sex again.
Okay. So yep, Myles is here, so I'm just going to start the segment early. So, reminder guys, just link down in the doobly-doo description to donate or contribute to the experiment.com. I actually forgot to mention that if you do become a contributor to the project, you won't actually be billed until the project actually reaches the target. So if you donate and the project doesn't get funded, you will get the money back. You won't actually contribute anything. And if you do actually contribute and the project does go ahead, you will gain access to lab notes that I post online. I'll be posting updates about the research. So basically you will find about how the research is going before anyone else, before publication, when the publication is out, and probably other amount of other goodies that I will be able to think of that I can do. If you guys have if you guys have any suggestions about treats or little things that I can do to encourage people to donate or contribute, please do let me know.
Well, I was wondering if we donate a certain amount, do we get a credit on the paper?
That I'm not actually too sure. I think you might get a mention if you donate a certain amount. I'll actually have to confer with my supervisors on that one. But
can we set up things that if people don't donate will kill them or severely hurt them, because extortion works?
I think that's illegal in my country, but you know what? I think it's legal there, so work away.
In Northern Ireland, it's just a way of life.
In Northern Ireland, car drives you.
No, in Northern Ireland, car drives over you.
Touché. We'll be back in a second with Myles, so talk to you then. Hello, yes, hi, sorry, we're going straight through live because I'm doing this on Xbit and I don't want to kind of break it all up and make an absolute nightmare, because I'm afraid my laptop is actually going to break down at the sign of any bit of difficulty.
Oh, blimey.
It's one of those kind of difficult laptops.
God, face. Oh. How are you doing anyway? You alright?
Yeah, I'm doing okay, just a bit brain dead at the end of a first day. I actually had a lab meeting and everything that went on way too long.
I know what that's like.
Okay, so hi guys, welcome to the third and last hour of this Trolling with Logic special. I'd just like to welcome our guest Myles to the show.
Hi there.
And
hello.
Hi.
That's right. And Mortimer and Ziller as our co-hosts. And the topic we actually are discussing is bad science, isn't it? And sorry, I'm just miles away at the moment. Bad science and dealing with homeopathy, anti-GMO, and sorry, I'm just everything's coming in at once at the moment. And of course, so yeah, anti-GMO and anti-vaxxers.
Cool. Where do you want to begin then?
Well, maybe we should begin with, I think, anti-vaxxers. I think that's maybe probably the kind of the most, probably the one most people will be more familiar with the bad science campaign around on this channel, the anti-vaxxer movement. Probably a good question to ask. What kind of brought the anti-vaxxer movement to your attention, or what's made you decide? What you made videos about the anti-vaxxer movement before? What kind of inspired you to make videos about that particular brand of pseudoscience?
Well, it was actually the first video I ever made was Sorry, get my camera sorted here. It was actually the first video I made. Was one bashing people who were against vaccinations, and I guess it really comes from when I was at university. Mumps was a serious problem in Manchester. We had entire halls of res that would just be, all infected with mumps, and this was crazy. This was back in like 2006, 2007, and it's a disease that really shouldn't be affecting as many people as it was. And then I knew that in the news a lot of stories had been about anti-vaxxers, but I always thought there were a very small number of people. It wasn't until it became such a massive problem in Manchester that I was like, oh, I'll read a bit more. And also, I not to go into too much detail, but I know someone very close to me who was affected by it. So yeah, I really had to be under my bonnet about anti-vaxxers. I mean, it's something. I always say there's something very almost lazy about certain people who I know it sounds awful, but it's kind of true, who don't look into detail about stuff. They've heard that vaccines are bad off the news and whatever, and they won't go any further. They won't talk to their GP. They'll just say no, they don't want to take the risk because they don't see polio anymore. They don't really see mumps and measles until, well, like in, I guess, was it 2013, where suddenly measles came back, and then we had scares, particularly where I'm from, which is in the northeast of England, a place called Middlesbrough. It was the place that was pretty badly affected, that in here in North Wales. So yeah, my first video was on it actually. It was from a guy. It was about a guy who was from Birmingham, who was saying, "Don't get your kids vaccinated," and he had 4,000 viewers, I think. And at the time, that was like a phenomenal amount of people. And I thought, fucking hell, I have to do something. Oh, can I swear, by the way? I just realised.
No, it's okay.
Oh, yeah, go for it.
Oh, we've long forgone any no swearing laws, any no swearing policy on this show.
Awesome.
So.
Right. I'll try and cut it down anyway. I get told off for swearing, like by my mum of all people. Yeah. Anyway, so yeah, I made a video where I basically called out everything that he said. He was going through the lists of ingredients that were in vaccines, and he was saying, "Look, this is just awful." And I went through point by point, being really quite anal, discussing every single one, saying why he was wrong, why it was in there. And I uploaded this video, thought nothing of it, and then I checked back a few weeks later, and I couldn't find his original video. And I was like, oh, where's it gone? And then I found, because it was back in 2010, they used to have a sidebar where you could describe what your channel was like. And I found out that he wrote something on the lines of that he's changed his views on vaccinations. I was like, oh, bloody hell, that's fantastic. There's real power to change people's minds here. And that's really where I started on YouTube. But yeah, anti-vaxxers, they do annoy me, because I find myself arguing with people in the pub all the time about it who don't get their kids vaccinated. And I'm like, please, honestly, it's totally worth it. When they're 20 and they don't have to worry about measles, mumps, and rubella and all this kind of jazz, they're really going to thank you. Otherwise, if they get infected by one of these, you can almost say goodbye to grandchildren and stuff like that. They're not nice things.
Yeah, that's really a bit of a particular reason why I don't really like the anti-vaxxer movement, because it doesn't really affect the adults. It's more of the kids that are going to be feeling the brunt of this, the kids of the anti-vaxxers, and not really the adults themselves. As you said,
it's not just them. That's the thing as well. People, we don't live. People presume that we live in a perfect world where everything is going to work 100% of the time. It doesn't. Vaccines do not take sometimes, and also we have people who can't be vaccinated because of medical reasons. So by getting your kid vaccinated, you are protecting them.
Yeah.
I think that's the big thing, isn't it? That by not vaccinating your children, you're also putting other people's children at risk. I think that's the thing that probably annoys me the most, although obviously neglecting your own child's care is bad enough, but to then neglect the care of the society around you is very dodgy. And
this whole, as far as I've read into it, it really kind of kicked off this new anti-vaxxer movement through Wakefield study, which has been absolutely that's just an awful.
To be fraudulent, or
was
it just bad?
Yeah.
No, it was fraudulent. He made up data, didn't he? I think.
He tweaked it to the point where it's not really recognizable. So you can tweak and you can say things, and if you're not, I think Concordance did like a fantastic video on it before, where he was looking at PCR. He did something there, and he said like, this is the same. You can tweak it past the point where it actually is what it originally was, and that's what he did. And he also made these kids undergo procedures that were totally unnecessary. So he got absolutely smacked down for that. And also his paper is a lot of people haven't even read his paper. It says in black and white that they haven't shown a link between autism, certain bowel problems, and vaccines. It says it's like a very preliminary one. It wasn't until the press conference where they actually said they found this link, and people seem to remember that and forget the paper.
Yeah, but that's the kind of the key. That's really key for any really pseudoscience that cites a peer-reviewed paper, is that generally they haven't read it. Such as that famous anti-GMO paper by Solani, which
the Roundup one.
Yeah,
Yeah.
I remember reading that, and I've never felt such a weird confusion of absolute rage and confusion, because it's just really, this is the paper that people are citing. No. And there's also other papers as well, like that have been cited by creationists, that don't say what they're saying, or they might read the abstract, get a gist of what it says, and then says, "Oh, this supports my idea," which, if you actually read the paper, it honestly doesn't.
Should I tell you who's the absolute... Oh, sorry. I was just going to say, the worst people I've found for that are geocentrists, who jump on things about cosmic microwave background radiation, and they're talking about the strange axis of evil, the dipoles that are all supposedly aligned, even though they're not. And they do that exact thing. They just pull out things from the abstract, put it up, and say, there it go, it proves my point. And you read through the paper and you go, actually, they're saying the complete opposite to what you're saying. People don't seem to read the papers that they're citing half the time.
Yeah, it's true. It happens all the time. I mean, I made a video a while back about one looking at Bt delta endotoxins, where it said that it would cause leukemia, and then you look at it and it's not specific for Bt toxins, it's for the bacteria, and you think, well, those are completely different things. I once had a guy give me a paper talking about the money side of getting people circumcised, and I read it, and I was like, okay, I don't really care about the money side of things. That's really, to me, irrelevant. It's all about, you could argue eugenics from that side, and that's not, well, extreme eugenics from that side. And he's like, oh, no, you're just reading the conspiracy. You're just thinking it's all about money. It's not about money. I was like, you just sent me this fucking paper. This is the paper you've sent me. I took the time to read it.
Actually, I've responded to an E-Juby video quite recently where he actually takes a New Scientist article which says that reports on a recent discovery on, well, I'm not sure how recent it was, I can't remember the actual year it was published off the top of my head, but it was on XDNA. And New Scientist had a little paragraph where they said, oh, this might help us to understand what early life might have been like, or this might help us to understand what life might be like in the outside of our planet. And he'd taken this little piece this little paragraph by New Scientist and blew it up to say that the scientific community believes this discovery is proof that this discovery proves that life, that they can prove abiogenesis, and that life, and basically made about abiogenesis. But if you actually read the paper, they never made any such claim. It was only really about reporting about how they actually made this molecule.
The best ones have to be the ones that go and quote undergrad research and say that, hey, scientists believe this, and you go back and you're like, oh no, this is just, a third year BSc student who said this. Oh no, the scientific community totally behind this person. All right. Not saying that you should flat out dismiss any undergrad research. I wasn't slagging anyone off there. Sorry.
I'd hope my undergrad research wouldn't be out there. It's basically very kind of basic research. That's crap.
You notice a really huge difference in quality between just the bachelor level and the master's level.
Yeah.
And then when I compare my master's thesis to my bachelor's thesis, it's you can tell there's like 2 years of development between them, and that's still nothing compared to what a PhD scientist would write.
Then there's a whole lot to go in there between bachelor level and masters, and just in terms of writing. That was actually one of my biggest pitfalls. But sorry, I think we're going off on a bit of a tangent on that one. So that's the actually select. What I've also noticed about pseudoscience is that they tend to use very questionable sources as well, from sources that are not intended to be scientific authorities, such as the Daily Mail, or well, that's an absolute authority on crap, but popular science magazines, and saying that this is what the scientific community believes.
Yeah, creationists love the National Geographic, don't they? And now on the age of the internet, where anyone can write a blog, usually people just quote blogs now, who quote other blogs and other blogs, and it turns into some massive circle jerk. And quite often, there's no recognizable research at the end of it. I've found it's a fun little game though to try and track it back to its source.
Sorry. Sorry, I really need coffee at the moment. I never go 2 hours without coffee. That's a general rule when you're in science. What I actually noticed myself was when I was actually going through one of the one of the scientific databases, I think it was Web of Knowledge, is that some of those databases actually have homeopathy journals as well. Of course, there's only really one that I know of that's science. I'm not sure my quotation marks are being seen there because they're actually very tiny on the monitor. But sorry, I have no idea how to fix that. Are there? How would you be able to tell for a layperson what would be a good reputable source from a disreputable source? Something that's, how to separate the good science from the bull crap.
I get this question all the time, and unfortunately, there's no answer to it. You have to know your field. You have to know what you're talking about to be able to decipher it. I mean, it's like if I were to jump into computers or engineering and I looked at some research papers around there, I just wouldn't have a clue where to start. I mean, I know probably to ignore the ones that say that the Earth is hollow and that gravity doesn't exist, but like apart from that, I wouldn't have a clue where to start. So you really need to know what you're talking about. Unfortunately, there's no easy answer. You just need to be able to get your teeth stuck into it, unfortunately.
And that's what I very would agree, because science can be very methodical, very precise in its language, and then there's can be a lot of jargon, but it's still necessary jargon that can put off a lot of people who really won't be into that field. Even different fields of biology, you can be able to confuse. For example, I won't be able to go through a genetics paper or a paper on bioinformatics that easily. Whereas someone...
I tell you what I love, sorry to interrupt, have you guys seen the cartoon that's going around now that says like movie scientists versus actual scientists? And they've got one where it's kind of like the movie scientist is like, oh, well, I'm an astrophysicist, but I can read this genetics paper and get you your dinosaur in a couple of minutes. And then it's like, oh, the real person's like, I'm a microbiologist and this paper's on bio whatever. I'll better ask someone. Because yeah, honestly, scientists are incredibly specialized. I mean, me and James on a podcast I do called League of Nerds, we always say that it's a special trope. We call them doctor scientists because they just turn up and they know everything. Yeah.
Like, there's a video you did not so long ago, Myles, where you actually asked, was it James, a few questions on chemistry, and he was just hopeless at almost everything.
I felt so bad. Like he was also really hungover, bless him. We had 2 nights on the trot. but I'm really hoping that I don't get asked any biology questions. He's going to spring it on me one of these days, and I have so many people being like, "You're a dick for doing that," but he's going to do it. I know, and I'm just going to look like a fool. I deserve it though, to be fair.
That could be a fun little drinking game. every question you get wrong, shot. At least you'll enjoy the experience.
Sounds like a good game.
Don't be giving these guys ideas. You know, the fun game for them is get the Irish person drunk. Actually, just on the doctor scientist, there's actually one person that I would be more familiar with. If you ever heard of John Pendergast, or what's his Penders, God, I can't pronounce his second name. He's a creationist, but he's claiming that he's a chemist, even though he is a auto mechanic.
Pendleton.
Yeah, he's
Pendleton.
Pendleton, yes, John Pendleton.
John Morris Pendleton.
John Morris Pendleton. That's where the Pendleton.
Tell me more about this guy. Go on then.
Logic actually did a 13-part series. I'd recommend watching. But basically, he appears in. If I actually could get my lab coat, I could probably do an impression, but. I'll probably leave it for the moment. If guys, if you can donate, let's say donate maybe €20 to the people in the chat. If you donate a certain amount of money, I will get my lab coat and I will do an impression of John Pendleton. But basically, he's an auto mechanic who basically does a creation seminar. I actually prefer to leave respect for the word seminar or conference.
Yeah. He has not understood what a seminar is.
Yep.
He just stands in front of a camera and talks.
Because it's a sciencey sounding word. And he calls himself scientist chemist. Who's an
chemist?
Yep. Pretty much.
Back, I'm scientist chemist.
Yeah, I've literally never heard anyone introduce themselves in any serious way as I'm a scientist. You know.
And his credentials are so dodgy as well. I mean, he did chemistry at undergrad level, then came out, did a year's chemistry in industry somewhere, and now he's an auto mechanic, and he thinks he understands the intricacies of biochemistry.
Biochemistry, biology, and all those sort of things. Because I did a degree in chemistry. That means it's a science degree. That means I can science on everything.
Oh, that's kind of hurtful. Why are you slagging off chemists? Jesus.
It's not really a real science anyway.
It's not really real chemistry.
I'm not having biologists tell me. No, I'm kidding. Sorry.
Well, I'm a biochemist somewhere in there. I'm a weird kind of field. But I don't know. Chemists are okay. It's those engineers that I'm. It's Cal around.
I always think chemistry though is the one difficult one to talk to people about, because it's essentially glorified cooking. So whereas biologists can talk about interesting cells doing all sorts, physics guys can talk about loads of interesting stuff about the universe. Chemists are like, well, I can make one white powder turn into another white powder. I don't know. Especially if you're organic chemistry.
It's the drugs trade, isn't it? That's what you've got. You're a chemist.
I have often thought, if this whole chemistry thing goes tits up for me, I can always make meth. So there's always that.
I'm already living the dream. I mean, I've said too much. Your Honor, I have no meth lab here.
Or Kitch disappears to snort something off the counter quickly.
I just have a cold. It's cold here. I have no heater.
So go on, tell me more about this chemist guy. What does he say? What does he talk about?
Typical creationist bullcrap, but he actually wears the lab coat at the actual seminar.
Oh no, that's right. That's a red flag for anyone out there. If anyone wears a lab coat and they're not in a lab, then you should ignore what they're saying. And if they're not, this is another one as well. If they're on the TV and they're in a lab and they're not wearing a lab coat, it usually means they're really high up. So those are the two things. But otherwise, just don't listen to them. I mean, why do you need? You don't. In fact, you don't do that. It's like safety 101 to keep all your stuff within contained areas.
Yep.
But if you're not
I've
wearing a
got
lab coat, how do we know you're sciencing?
Because you introduce yourself as Doctor Scientist Chemist.
touche. In fact, I've been yelled at many a time for actually wearing my. Well, it was more over somewhere else for wearing my lab coat outside because you kind of get used to it. But that's just. It's so sad that I actually thought he was a parody.
Oh.
Just as soon as I saw the lab coat, I was thinking, really, you're trying too hard.
But he doesn't help himself because he goes on about UFOs, and in one with, is it Charles Bauer or Carl Bauer? He goes on about an expedition that he's planned that nobody's heard anything about since, where he was trying to find a live pterosaur somewhere in South America.
Oh,
Wow.
God.
And apparently people have been known to die from pterosaur breath.
Oh. Good to know.
Like,
I
want to see
I've
that
no
autopsy report.
I have no idea how to respond to that. Actually, there's actually another. Actually, bringing up him actually reminds me of another pseudo-scientist that you guys may be aware of. Actually, Mike Adams.
Oh, I love it.
Yep, good old Mike.
He's amazing. He was on my radar for a bit, but when he really came on my radar was when he was saying that the government was putting sarin toxin, nerve gas, whatever, into the water supply to fluorinate it. And that's when I was like, wow, you're a legend. And then, of course, his rap at the end of Seeds of Death, which is amazing. What a legend.
Yeah. And that's actually one of the reasons why I'm trying to push towards us doing a live ponage of that video sometime this year, just so I just experience that live and to get everyone's reaction to that, because I think that's both a weird combination of cringeworthy but epic at the same time. It's a weird sort of thing.
It made that movie go from like a 1 out of 5 to a 5 out of 5. It's amazing. If anyone's not seen it, then you should totally watch it. It's epic. I think there should be more rap videos at the end of anti-science documentaries.
Certainly make them more interesting.
I loved your podcast, by the way, about the music in certain things. I think it was 9/11. They have like a music scene.
Oh yeah. That was quite a fun podcast to do, actually.
Have you heard of the chemtrails music scene? That's the one that's really, really quite big. The 9/11 one's alright, I've heard of it before, but the chemtrail one, have got music videos and everything. It's really quite professional. It's really strange.
I have to check that out, actually.
I have to not check it out.
You don't know what you're missing.
But actually, just about Mike Adams, he actually has or claims to have his own lab where he tests different foods. He actually claims to have bought a mass spec.
I could just imagine him putting like a sandwich into a mass spec and hoping to get back something. What does he plan to do with an MS?
I think he's looking to detect different. I think it was metals. I think he was looking for heavy metals in food. I can't remember which one just off the top of my head. Cal will be more up to date with Mike Adams, because I generally since I think it was halfway last year, I've kind of avoided him just for my own mental health, because there are some topics that just make me go absolutely furious.
Like what?
Actually, funny enough, I've tried to actually do a video response to his webinar series on YouTube about Ebola. I got halfway through, I was already sweating with rage, because you can imagine what he was saying. Don't go to the doctors are trying to kill you, this kind of typical stuff. I was working my way through it. Next thing, computer went. My computer just crashed, which I assume was from the idiocy, but I took it as a sign that Michael just don't. Give it a break.
It's really awful though, isn't it, that all these people have jumped on the crazy Ebola bandwagon. I mean, I think the Daily Mail published something in December about homeopaths going there, and it presented them in a positive light. Did you guys read that?
That one I
I
haven't
didn't
read.
know.
About it.
It might be in the Mirror. I can't remember. One of them, yeah, was going on about these 4 homeopaths that came from all over the world, and all they wanted to do was treat Ebola, and how the World Health Organization said they're not allowed. And it was almost preaching. It was almost saying that these people were doing good. And it's like, oh, Jesus Christ.
Yeah, but if this is the Daily Mail, you know, they've like,
You could say that homeopathy is treatment for Ebola now, technically. So yeah,
keep the system flushed.
yeah. But
Yeah.
I hear as well. I've got a particular bee under my bonnet under. I keep saying that bee under my bonnet for a guy called Robert Scott Bell, and he's done a thing on Ebola as well. And his sounds exactly the same to what Mike Adams is saying. Don't go to the doctors. You can treat it with antioxidants and crap like that. I mean, luckily, like there's not going to be hardly anyone in America who's got Ebola or anything like that. It's really quite difficult to catch. The problem comes when, you know, now with the age of the internet, that kind of information can leach over to West Africa, and then you have lots of problems. I mean, I know you guys talked about it before in Trolling with Logic. There was the blog post that was too nutter butters even for natural news. The guy who was saying that you should make your own Ebola homeopathic solution. That is
Terrifying.
flipping crazy.
Yeah, that I remember coming across that post, and I was just, I had no idea. It just takes a very special mind to be able to think that's a cure for Ebola. Is basically, you know, infecting people with Ebola. That's where that was going to end up. And it's not like and also, yeah, it's a very real possibility that this stuff will get over to the affected areas, you know, Sierra Leone, and you know those places already have enough trouble with the mistrust of you know, with the medical.
Yeah, I mean,
help.
a lot of people presumed that it was Western people coming over and infecting them. In fact, there was in November, October, there was one of the clinics was raided, and they actually took people in blankets that were covered in blood out, and that was a serious problem.
Yeah, I haven't heard that. I actually didn't actually hear that, but that's you know, awful, and that's why, you know, it's important to try and get out accurate information and dispel the misinformation that's going out there, because people are going to act on this on this information, and that's also probably one of the responses actually to the questions is, what is the harm? You know, when it comes to, I'm sure you get it, and I'm sure Martin and myself get it as well, is actually what is the harm of the pseudoscience that you are debunking? Why can't you just let them have their little parade? Or
Why can't we let them believe what they want to believe?
yeah, exactly. Is that people are going to act out on this information, and in the case of Africa, as you mentioned there, you know, people aren't going to get treatment. People are going to do things that are going to help the disease spread, and with a disease like Ebola, you know, that can be very, very dangerous. And also,
I think, sorry, I keep interrupting you.
So no problems. Sorry, I'm just kind of trying to fill in the dead space.
I was going to say, I think a main problem with these people producing stuff is that they think that there's not going to be anyone at the other end of the line. Or if they do, they think it's just going to be people who need them that are going to buy their products. They don't think that it has the potential of doing real harm. Well, maybe I just want to believe that, is what I'm saying. I can't believe that some of these people can be so... I don't want to say evil, but dangerous. Careless, aye. I don't know. It just winds me up. Sorry, I'm mumbling.
No worries. I'm probably going to be doing that for this whole show. But yeah, it's. But like, there are people out there that genuinely genuinely believe in what they actually promote, such as spirit science, or I think.
I I don't know if he believes it himself. I don't think he cares if he believes it or not.
How come?
Well, he just it's like he's promoting so many things that are mutually exclusive.
So he's contradicting himself.
Like in his first video, he says that kids are starving in Africa because of the Western world keeping resources away from them.
Yep.
You can't believe both those things.
So, do you think, like this guy in the comment section here says, that he's doing it to make money?
No, I don't really think so. I think he just... I think to him it doesn't matter if it's true, because all that matters to him is that it feels nice.
I think also that it promotes him.
Of the truth is completely irrelevant to him, I think.
I think I know what you mean. It's more, he's saying it because it feels good to him, and that it gives him a nice feeling. It gives him a fuzzy feeling.
Yeah. I've not watched that. Sorry, I keep interrupting you again. I'm going to stop it.
No worries.
I've only watched a few Spirit Sciences so far. They're like a fucking hour each. Do you think there's real harm in them? Because if he's saying, "Oh, it's the Western world getting down Africa," is that really going to do? That's just rambling. And the ones that I've seen so far, it's just words that he's throwing there. It doesn't seem to be anything too bad. I mean, you've made a whole video series on him, haven't you? So, do you think there's real danger behind what he's saying?
The stuff about medical advice.
Oh, I haven't seen any of that yet.
No, he's basically he's saying things like, okay, if you feel depressed, if you have anger issues, you should just meditate.
Oh.
Don't go see a shrink. I mean,
doesn't he also put down children as things that are a bad influence on our lives that we need some kind of we need to redress the balance from or something?
No, that was also about the meditation thing. Like, whatever your problems are, just meditate and that'll take care of it. Don't actually address the problem. And among the problems, he lists children.
Yeah.
What you mean, children with problems or children as problems?
I don't think he means that children are a problem. I think he means like the problems that your children might have.
Oh.
Don't address those problems because I think the idea is this sort of pseudo-Buddhist. Your desire to not have problems is the source of your problems, or something like that. So just be okay with problems and you won't have any.
Because they're fantastic, aren't they? Really, for YouTube.
Yeah, I mean, the work he puts in. A lot of people will say that, well, it's just a few hours in Flash, and yeah, but there's artistic talent behind it.
Oh, I forgot. I was actually listening to one of your podcasts again today as a bit of prep for this. You did briefly talk about it. I remember this now. And you talked about the Smiths.
Yeah.
They're amazing. Have you seen some of the tweets that they put out? They're absolutely...
I've read about a lot of it.
Do you not think if you're Will Smith, you probably take away the phone from your kids and just say, listen, you'll thank me when you're older because this is going to be with you for quite a while?
I think his money has gone to his head. I think he just... I don't know.
What about some of the stuff he was saying that you don't need school, or people would be better off if there was no school, or something like that?
Well, yeah, school isn't true because it ends.
And if babies could talk, they'd be the most intelligent thing in the universe.
And the time doesn't exist because you can perceive it at different rates depending on if you're having fun or not.
I think that
But
he
that,
is.
I actually contemplated making a video addressing one of their interviews, but then it just sort of it dawned on me that they're 16 and 14, I think.
Yeah,
And
they're kids, aren't they?
Yeah, I think
yeah,
no one is
they're
actually.
kids.
I think no one's actually going up to him saying, "Okay, defend that." You know, I know as a kid, if I said something stupid to my parents, they'd be asking, "Okay, what the fuck have you just said?" You know, they're kids; they're gonna say stupid crap because you know they don't have the life experience. They actually haven't done any to study. They're still going through school. But Smiths, you know, I don't think anyone is there to actually say, "No, you're wrong," or try to give them critical feedback on some of the ideas that they have.
I think a lot of that has to do with the fact they've grown up in an environment where they have everything.
Yeah.
When you have everything, you don't have to think. You don't have to be intelligent. Case in point, Paris Hilton. I mean, it's the same thing.
Especially when you grow up in an environment that's filled with sycophancy, which, you know, is going to be a lot of their environment in Hollywood anyway.
Mm-hmm.
Because the lack of telling them when something's crap. I mean, how else do you explain After Earth? Oh my god, terrible film. And also, I don't know about you guys, but I am so grateful that the internet wasn't around like it is now when I was younger, because Mini Myles was a right dick, and like I would hate for any of that crap to still be around right now, my stupid haircuts and all the rest of it. Yeah, so I understand what you mean. Like these are just kids, but that's why I think Will Smith should take their fun away from them and be like, listen, you know, you're gonna regret this. You've been a silly teenager.
Yeah.
But I just couldn't believe some of the stuff that they were coming out with, and I can imagine the look at the guy who's there managing their PR. He's probably in the hospital right now, just recovering from the stroke he had.
I wouldn't be so sure. I think.
Oh wait, was that a joke?
It gets attention.
No, that was actually a joke. Sorry.
Yeah, I just for a second thought, "Fuck, has he just had a stroke?" And I'm like laughing at that.
No, I won't be that evil. Yes.
So, what are your guys going through your? What's your one beef that annoys you the most? Then I've talked about mine. Definitely has to be the AIDS nihilist. But to go through you all, what's the one thing that really just gets on your tits?
General science denial. It doesn't really matter which specific subject we're talking about. Just the fact that people seem to be mistrusting science, and they refuse to learn about it. They don't want to understand it.
For me, I think this is cheating because it's kind of. I think it's kind of encompassing a lot of different ones, but I'm not sure how to phrase it. I think the best way to phrase would be the New Age medicine movement. You know, where they kind of are pretty much like germ theory denialists, AIDS denialists, anti-vaxxers, that kind of crowd. You know, those that really argue against kind of, you know, the bad medical science bingo. They check all the boxes. Those kind of people really just get under my skin. They're just, I think they're the most dangerous, and that's why they really are the bee in my bonnet on that one. And some sections the anti-GMO movement. I mean the obnoxious kind of segment, but that's just because they're obnoxious. No, I mean the obnoxious ones, not just anti-GMO in general, because some of them are actually quite friendly, I found.
Oh, I found that. I've talked to a fair few of them. People are lovely anyway. I find, like, even the most batshit crazy people, as long as you don't talk to them about their batshit crazy stuff. Yeah. And what about you? Finally, what's your thing that annoys you?
Well, I guess I'm involved in a bit of environmental kind of activism and stuff, and the thing that really annoys me is the amount of pseudoscience that creeps its way into that whole section of, I guess, society. In the environmental activism movement, you have a lot of these really strange anti-GMO views, anti-nuclear views that they take away from real environmental concerns. They push the discussion into this really unuseful area, and take it away from from what it's actually should be about. And a lot of people involved in the activist scene who don't really understand the science at all and don't know what they're talking about, so they end up coming out with something that's complete rubbish. And it's a bit of a bugbear for me, being semi-involved with that movement and yet seeing so many people who are they're kind of a mouthpiece for a lot of movements, coming out with just the most ridiculous, inane rubbish that they get down to the bottom of it is stupid.
I'm in an environmental science lab, and my supervisors all the time, whenever you think about that, these people, can we just ignore these people who are just... I'm trying to censor it down just so I know he was on a couple of hours ago, so I don't want to kind of paint him with a bad brush. But that sort of stuff myself and himself can get into awful rants about, just back and forth complaining about it. And especially with stuff like bad science and anti-GMO, actually is really bad because, for example, let's just take an example there. A couple of years ago, there was I think it was I can't remember. There was a proposition in California. 37, yes.
I was there at the time. It was all going on.
Oh yeah, I think I remember. Did you do a video about that, or am I?
Briefly, yeah. It was an advert I did for it. Yeah.
Yeah, I remember that, and that was a really good one. And sorry, I don't mean to be kissing ass on the screen. But I basically read with you where the anti-GMO people, it's my opinion that the anti-GMO people had did such a bad job at the science and relaying what the science is like and giving themselves a bad name, that this proposition didn't pass. And it was a decent proposition to label that.
The more I read about it, the more it was a bit dodgy. It wasn't just the labeling of genetically modified food. It went even further into kind of pushing other stuff forward, and that's where, because I didn't read that much into it. I was just basically, right, this is labeling food. That was the main thing in my video. I'm fine with that. But yeah, it turns out it was a lot more than that. Yeah, I didn't know. Mind you, it's not my country, so yeah.
And I was over here, I can't remember actually where at what point I was at. I think it was at my final year at that stage. But the anti-GMO people, they're giving themselves such a bad name when they're quoting papers like the Serlani paper, or even just going off the walls with citing, getting people like Mike Adams involved, or... Well, I don't want to say the anti-GMO crowd is quite a big. I don't think most of them want to be put up with Mike Adams, but these are the kind of people that are coming out and that are seen.
The worst thing about them, I think, is that the majority of them aren't just fighting it against GMO food; they're fighting GM technology as a whole. So, as a microbiologist, I'm guessing you've done a lot of that, and. One of the best things that we haven't published it yet, though. Me and a friend James went to a March Against Monsanto protest, and he's talking to someone on camera, and he's like, "Okay, what do you want?" And they said they're against GMOs. And he's like, "Okay, well, I'm a scientist. You will put me out of a job. I'm doing X, Y, and Z that has the potential of doing blah 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 blah. Do you want to stop me?" And these people are just absolutely taken back, because they don't think. One thing I was amazed. When I talk to these people, is the lack of knowledge they had, and I don't mean that in a really big-headed, arsey kind of way. I mean, these people honestly didn't know why they were there. They heard that GMOs were nasty and dangerous, and they didn't like the companies, and they'd seen pictures like in the Seralini paper, and they've seen all these kind of like fake paper. I'm going to say, yeah, they are fake papers. They're not real coming out, and they were quite scared. I mean, and that's the thing. They weren't bad people. They were doing something that they thought was right, but they were so uninformed. Yeah. Oh, and Michael, there's something here that might really annoy you. There's a comment here that says that someone went to a cancer support seminar with his mum, and they said there was a lot of woo on display there, and it was terrifying. That's awful. That when you think about it.
Yeah, especially when it's a cancer support seminar.
Yeah.
This is literally like, it's not just on YouTube or, where it's just, you're not really going to be thinking you're more. I'd say that's more careless. This here is at a cancer support seminar. Is just, I want to stop. It's just bad. It's absolutely disgraceful.
It's almost like vultures, really, isn't it? That's a horrible analogy. Just like preying on. No, that's a terrible analogy. Take that back. Sorry.
Yeah,
I don't know. Is it that bad an analogy?
I don't think so.
Maybe not.
But it's an absolutely terrible thing, and especially, and I will even step that up because I wasn't too sure if they were just there. Oh, this is what we believe, blah blah blah. But they were actually selling. But I assume probably they were selling books and pills and all that kind of things. That seems to go hand in hand.
One that
That's
I don't
evil.
get is ozone therapy. Have you heard of that before?
I have never heard of that.
Yes.
Is that where you breathe in pure oxygen or something?
No. Ozone, so O3.
Oh, right.
So.
That sounds like a pretty stupid idea.
Yeah.
I was gonna say, that's poisonous, isn't it?
Yeah, you'd undergo ozonolysis, which is breaking of carbon-carbon double bonds. So it would just dissolve you. And they recommend this to people with lung cancer. And you're like, wow.
That is
chemist.
That is a very, very, just for the record, that is a very, very bad idea.
I know, like, ozone buggers over all sorts. So it's unbelievable. You can get ozone chambers. What's really awful, if you look on YouTube, there's actually a video of a guy trying it when he had cancer. Well, I think I didn't watch until the end. So it's just, yeah, it's awful.
Yeah, that's gonna absolutely destroy the lungs.
Well,
there's a one key in cancer therapy is try not to kill your patient. It's good to know. That's probably the golden rule. Is it's alright if it kills the cancer cells, but if it kills you, what's the point?
There's a group I found on the internet once who were people that thought drinking hydrogen peroxide was good, and their thinking behind it seemed to be that H2O, it's got oxygen.
Yeah, an extra
H2O2 got more oxygen. That must mean it's good. And they had no idea of what chemical bonds are and what happens when you add another oxygen atom to that chemical bond, how it changes the chemistry of the molecule.
Have they not seen that cartoon where it's a chemist and his friend go to the pub? He goes, "I'll have a H2O," and the other one goes, "Oh, I'll have a H2O2."
Yeah.
That was just thinking that one there. That was. But yeah, there's an awful lot of wacky cancer treatments out there. I know there's one actually done by a guy, well, it was Kent Hovind, but the first time I came up across this particular treatment was through Eye to Eye. I'm not too sure if you guys remember this particular person, but he said that the vitamin B17 in the pits, in the seeds of, I think it was almonds or pears or apricots, I can't remember. Peaches, that was it. That could cure cancer, but when you actually look at it, the only thing you're probably likely to get is cyanide poisoning if you eat enough of them.
That's still going to this day. People will take all sorts. Like it just bumps up the cyanide in their system. Not good.
That one's a particular. That one was a particular B. Sorry, I'm following you on that one. That one was just a particular annoying me. That was a very annoying for me to kind of address that kind of thing, especially when it came from.
The reason I didn't like peaches.
The extra cyanide. Wrong.
Mortimer, is it? Wait, is your name? Is it Mortimer? Am I saying that right?
Mortimer.
What?
Mortimer.
Mortimer, sorry about that. Yeah, something I went to that I think you might like. I went to a new age hippie get-together meeting thing that was like a hotel near me, and I went to a talk that they gave, and this woman with a straight face, I think you'll like this, she was advertising smells that she made. No, wait, I said that wrong. She made like a load of scents, and she was saying it's for different things. You know, if you're feeling angry, you spray this. It's got gold in it, and you know, you waft yourself, and you'll feel stronger. And she says, "Oh, it's because it actually changes the DNA of you." And I was there, just like, "Don't laugh, don't laugh. They'll know. They'll know you're not one of them. Don't laugh." And then I had to stop from putting it down. Do you reckon I can commit a murder and then get some of this spray? No, don't ask her that.
That,
It's one thing that's intrigued me is I work a lot during the festivals, and you have these places at most of the festivals in the UK. They call them the healing fields, and it's full of like homeopaths, chiropractors, all these kind of weird little things. And one thing I've found is if you actually ever do need anything actually healing when you're at a festival, the last place you want to be is at the healing fields, ironically.
Because
you just don't get healed. You know, I've cut my finger open. Well, let's do a little bit of homeopathy on it.
oh, okay.
I think was there a holy shrine? I can't remember. Was it Lourdes? Sorry, I'm just kind of getting this back at the back of my head, but where they found that there was antibiotic resistant bacteria in the water.
No, sure.
So I'm just I remember actually coming across this actually just this morning on was shared to me on Twitter. I think it was New Scientist. But actually, funny enough, actually a past guest of ours actually was pretty much in India. The shrine. I can't remember the actual name. Sorry, I'm just awful at the names at the moment. There's a shrine in India, and that it was crying. It was one of these crying statues of Mary, and people were rubbing up against it, drinking the water, believing it gave them, cured them. Anyway, he came in and he was challenged. He was a bit of a skeptic, a bit of a famous skeptic in India, and he was challenged to find the source. And he found out that one of the pipes from the sewage system was leaking into the Mary statue, and that the people were actually drinking sewage water.
Oh,
nice.
So, and I think it was for that they pretty much kind of chased him off. But that was one of the past shows. I think it was 2 years ago now, actually. That one particular story has stuck in my head ever since. But
yeah, someone said it's like the Simpsons episode where Maud, there's the gas leak and the mask kind of floats up and they figure out what it is.
Yeah.
But that as far as I know, that those kind of things are pretty. Sorry, I'm just not getting along without my coffee today.
There, Michael?
Yeah, no, just having withdrawal symptoms at the moment. Fairly slowly getting withdrawal symptoms.
He's still recovering from New Year.
Yep, that too. That was one crazy night. But we've gone on to.
Oh, I wanted to ask a quick question, if I could, of Myles. Just on your HIV denialist series that you did on House of Numbers, you brought up the Foo Fighters who did the benefit gig.
Yeah.
Have they said anything about that? Have they retracted it? Or are they still adamant that they did the right thing?
I at the time I tried my hardest to actually find what happened, and all I managed to find was that they just simply removed it from their website. I don't think they've made a public apology. I read a few blogs and articles in papers that have been put online that said that the gay community in San Francisco was really quite angry with them, and how could they not put a retraction up? So I don't think they've done anything. I might be wrong, because this was like over a year ago that I actually looked it up. But unless they've done anything since then, I couldn't find anything. I mean, you'd think something like that would be massive and glaring on their website, like, hey, fuck it, we're sorry, we're wrong about this, we've caused probably a lot of damage, but it's not that.
Just a slow back pedal, but They probably still do, but I would say, just taking out that, though I'm not an absolute mind reader, so I'm only speculating that they still do in some way accept that, but there's just such a backlash that they decided to just quietly remove it.
I don't know. I would just be me speculating.
Yeah, no,
I guess maybe the death of Christine and her daughter maybe brought it home to them. Who knows?
It's sad the way that sometimes that, well, it almost what needs to take with some people, that it takes them to for them to personally be affected by it.
Well,
for it to sink home.
I think that happens all the time. Not personally, but like within close proximity. I mean, again, going back to 2013 when we had the measles scare. It's a result of people getting ill, and to be honest with you, I don't think most of these things are going to go away until we see another measles, mumps, rubella outbreak, until we see polio, possibly, even though polio is pretty much out now. But like, if we ever saw something like that coming back again in like the Western world, that's when people will get vaccinated. Because right now, you know, we all live in a pretty good part of the world where we don't have to worry about smallpox and crap like that anymore. So, you know, it doesn't seem. I guess living in this world, it seems more to risk to some people to get the vaccine than it does from the problems that they don't normally see day to day. So yeah, I think I can. It's not a pessimistic view. I just think it comes from a view of just more people are going to get hurt before it gets better.
I think that's also for other sciences like climate change. People aren't going to start accepting that until they start actually being really affected by it. I think there was a quote. We'll take it. It's all over the face. But actually, there was a question I wanted to ask, and that, you know, during your series on the AIDS denialist movie, you received several DMCAs. I was thinking, as far as I can tell, with the HIV crowd, that they seem to be the most. They tend to go after you the most, or they tend to almost bully in order to silence the people that are critical against them, more than, I'd say, spirit science or any other form. Do you find that? Do you think that's the case, or?
It's I think there was a little bit of orchestration around those people where you don't really get that with the other things, and I guess it's more. I think it more comes from AIDS denialism being under the radar for so long that they haven't had people being critical of them on YouTube and places like that because people haven't heard of it. So when I came along and was like, "Your movie fucking sucks," so do you. They took real offense to it. It didn't help that, you know, to be part of this. Little group, you know, you either have to be very unwell and denial, or a few other things as well. But also, it attracts the absolute nutters. It attracts the Liam Sheffs, the Brent Leons, and they are a few cabbages short of an allotment. So they all just came after me. It was actually it wasn't very nice. But unfortunately, what I've seen recently is this tactic of going after the person. This almost social justice kind of thing seems to be almost commonplace on the internet, no matter what side you're on. So I think it could even come from. I think maybe it could have came from just really the times that it's become more acceptable to DMCA someone, to go after them professionally. But I honestly don't know. They were very vocal about how much they hated me, though. they flipping hated me. Yeah.
That's generally that actually reminded me of back. I think it was a few years before that. I think it was 2010 in or around that. There was actually the big problem in DMCA was more with creationism, and that you had other big YouTubers being DMCA'd left, right, and centre. You know, the famous Venom Fang X. For those internet veterans who remember him going around.
It was a little bit different though with me, and with the creationists and stuff, it just tended to be one silly Canadian boy in his room filing all these DMCAs. But with me, it was like a massive company. They said they owned the property and they were properly coming after me. I mean, I know what I think would be brilliant, and this is what EFF are thinking of trying to trying to get legislation passed, is that if you file a false DMCA, there will be a second fine. And if that were to happen, then all the DMCAs would disappear almost overnight. Because even big companies won't risk like $5,000 a pop. I can imagine the businesses would come out of that. Say like you just have lawyers jumping at you saying, "Hey, you know what? I'll take 10%, and we'll sue this bastard because they filed a false DMCA." So if that happens, it's going to be fantastic. It's going to mean that the internet's DMCA problem is going to be gone. I mean, at the moment, I have 2 DMCAs against me right now from some lunatic who thinks that I can't use images and say they're under fair use from a book that also says they're under fair use. So yeah, as soon as that comes, that's going to be fantastic. And the great thing about it as well is the EFF are looking to say, hey, we're going to make sure so it's going to be backdated. So it means all these people that have filed them in the past against me, I can just go after them, which would be brilliant. And I honestly think that's going to help because the DMCA at the moment is broke. I mean,
it's an honor system, isn't it? Really,
Sorry?
It's an honor-based system. You file it under good faith.
Yeah.
Is that the current
But
system?
the problem is though that it's a way of getting information from the other party though, and if you deal with the kind of people I've dealt with in the past, I don't want to give them my personal information. I mean, look what happened, in February when they contacted my employer and they posted my stuff all over the place. These aren't very nice people. This isn't like a nice gentlemanly agreement in the pub between 2 people. No. Usually these people who wield it are nutters. And the worst thing about it as well, it says that the DMCA is filed under perjury and all that kind of stuff. But if I were to take any of these people to court, I could only sue them for the cost it would take for me to go to court and my losses. But as I don't make any money on these videos and therefore protected under fair use, they haven't really got that much to lose. Whereas I would have quite a lot to lose in taking them to court. So it's almost this circular thing that's just going to protect people like Liam Sheff and Larry Cook and all the rest of them.
Yeah, it's a system that definitely needs improvement, and that's actually can be used, and it's actually, as you said, it could be used for intimidation for people who may not be so willing or to give their personal information, who may not be in the situation to.
It's definitely used for intimidation, and you have companies that make a living off it. Psychic Sally's got another one who's been after me. She's DMCA'd me a few times. For those of people who don't know, leaning over. Psychic Sally is a psychic from England, who I'm currently reading her book, and it's as fantastic as you'd imagine. So she got a company called Web Sheriff to come after me, and they, again, not very nice people. They make a living out of censoring the internet, and it's pretty shitty, really. But like again, if this flat fee came in of $5,000. That company would be wiped off the face of the planet within a day.
And especially, it's not only just pseudoscientists, but it's also, as far as I'm aware of, actually creeping into the video game community with reviews of games being taken off because they're a bad review.
Oh,
so
is that a thing?
Yes, actually. As far as I know, actually one such example was, I think, one of Angry Joe's videos was removed. He was actually given the press footage and explicitly used, you can use this in your review. Next thing, gave it a bad review. Next thing was taken down.
Yeah, that's just blatant censorship. That sucks.
But yeah, But if you had a flat fee, or even an incremental fee, that the more copyright strike, the more times you falsely file a DMCA against somebody, the more you should probably pay. Because I think that will probably be.
I think people will be surprised at how often companies do this. I mean, if I got these 4 years ago when I first started, I would back down straight away. And it's just really shocking. I mean, these people are paid a hell of a lot, like web sheriffs out there, to almost to censor stuff that they don't like. They actually, I mean, they call themselves web sheriff. How fucked up is that when you think about it?
And basically they're at no loss. They are risking nothing.
Yeah.
realistically, they're really in the actual terms and conditions. They are technically they could be taken to court, but who's going to take? There's no one's going to take someone to court over a false DMCA, so they are risking nothing. So it's basically just
apart from the Streisand effect, it all goes tits up for them.
Oh yeah, that's actually probably the best weapon at the moment that can stop people from doing DMCA, but some people just don't know about it and just go press on right ahead. Or especially, but it's more. I find that would be more. The Streisand effect isn't as helpful to maybe the lesser known YouTubers who may not have much of an audience at the moment. So I think that the flat fee coming in that will do them the world of.
Oh no, I'm not saying it's coming in. I'm saying that's something that they're really pushing for.
Sorry, I mean, if it does come in, that's what I mean. Sorry, I'm just speaking.
I find it quite weird though that maybe I'm going a bit too extreme here, but that censorship in such a form isn't itself criminalized. I mean, it's the First Amendment of the U.S. Constitution, for God's sake, is freedom of expression.
But that only exists in the U.S. You could say that some of these countries and some of the people who act on behalf of them are from countries where that isn't a thing.
Yeah, but with the internet being such an almost U.S. institution as it kind of sometimes feels, I find it odd that it's not trying to champion the push against censorship probably in a more extreme way, but at least in a more, I suppose, a more pragmatic way. YouTube is just bending over backwards for people who file DMCA requests rather than actually pressing the point.
Well, it's all automated, and they have such a volume that they possibly couldn't do it, and also it all stems from when Viacom tried to take them to court, wasn't it? And how Viacom uploaded their footage to YouTube and then took them to court over it. So I can see where YouTube comes from. I mean, I know people in YouTube. I've been there, and they're lovely people. But they just want to protect their company and their interest. And the bottom line is, they don't really care that much. As awful as that sounds, they don't. I mean, if you disappear or if you get a few DMCAs against you, if you have a channel of 100,000 people, then even if you disappear the next day, that's just a blip on their screen. You wouldn't even register. So.
Sorry, just noticed we're coming to the end of the actual show, so at this point I'd just like to mention two things. You can still donate to the experiment.com project page. The link is actually on chat right now. It's in the description right below. At this point in time, I'd just like to ask, give Myles the floor to promote anything he may be working on, or the podcast itself, League of Nerds.
League of Nerds, yeah, with James. Yeah, I feel bad I can't promote that in your podcast. That's cheeky as hell. But yeah, I'm on the internet. Come find me if you like me.
And if you if you have a link, you could just put into the chat there, Myles. I'll put that for the podcast as well, because we liked it.
I think if I do that, my internet will crash. So maybe later I'll do it.
Grand. We'll probably put it when this video goes up. We'll probably have it in the description somewhere.
Cool. Well, no, thanks for having me on, by the way.
Cheers. Thanks for coming on. So.
Yeah, cheers, man.
Thanks for everyone who has contributed to so far, and here's to happy 2015 of podcasting.
Right.
What's the problem? There we go. And so we should be live. I'm just waiting for the broadcast to come up. Where's XSplit? I think it's a come up. Where's XSplit? I think it's a come up. Okay, grand. Well, are we on? I think that's a sign that we're on. Yeah, I can see us. Welcome everybody to this first Trolling with Logic of 2015. It's a very special 3-hour show. With us is Mortimer. How's your Christmas?
Oh, the Christmas was okay, but then I got the flu. So, yeah, I feel kind of like crap right now.
That seems to be going around a lot. And back on Ziller, welcome.
Hello.
And how's your Christmas?
I was okay, but I think Martimer's flew over Christmas, so I've probably given it to you for the new year. Sorry, man.
And a special guest for this hour is my supervisor, Dr. Enrico Marsili.
Hi.
And so this is a 3-hour show. The first hour will be just myself and Enrico will be discussing the project that is on the experiment.com page and be answering questions. And then the second hour we have Professor Philip Moriarty back on the show once again to discuss science funding. And then the third one is actually, I'm surprised we actually were finally able to get Myles Power from the League of Nerds. And Zeller is very overexcited to talk to him, as am I. I'm actually quite a fan of his. So, sorry, I'm still suffering from the Christmas fatigue. So, well, maybe we should start about the actual project itself. The project we have is about producing metal. The project we have is about producing metal nanoparticles, and usually the conventional methods are either expensive or use potentially harmful compounds to make these nanoparticles. We take a biological method, which are cheaper, cleaner, and just easier to produce. However, there are problems associated with this type of production. Mainly that the nanoparticle production is very slow. It takes days to produce these nanoparticles, which adds to the cost, and the nanoparticles themselves have different sizes, which adds to the cost because the properties of nanoparticles are dependent on their size. So we hope to add focus to the nanoparticle population, to the nanoparticles used by the bacteria Shronella, by adding in an electrical potential, and we hope to stimulate that bacteria using this electrical potential to increase the uniformity and decrease the amount of time needed. At the moment, it takes about 100 minutes as opposed to 2 days, which is a great decrease in the time. Enrico, do you have anything to add to that? I'm just a little bit.
Yes, no worry. I can take it for a few minutes from here. That certain metal-contaminated environment and sediments, including deep sea sediment, are able to interact directly with solid conducting surface like metals and electrodes. Basically, what's happening is that part of the energy respiration is channeled in oxidation of solid electron acceptor or donor, respectively. Shronella onedensis is one of these bacteria, isolated originally from shallow lake sediment in the 80s, and is capable of interacting with a variety of surface and material that can use as either electron acceptor or electron donor. This is the general microbiological concept. There are various applications for this direct interaction. The first is a bioremediation, aka the bacteria is able to reduce or oxidize metal from its toxic to a less toxic form, or eventually to precipitate outside of the water phase. In other cases, the bacteria is able to perform anaerobic oxidation, which are very effective for detoxification of organic compounds. And that's one of the applications. That application, although it didn't reach the peak that we were expecting, is the production of electricity. In a device known as microbial fuel cells. The idea was to convert directly chemical energy from low-cost substrate like acetate, lactate, residual wastewater, and residual biomass into electrical energy. Although this device didn't scale up to the dimension required for mass production, it is still a very good device to investigate the nature of microbial metabolism and for some bioelectrosynthesis process. The other application, which stems from electrosynthesis in Shionella, is the production of nanoparticles. Nanoparticles have been produced for years in biological system, but viable cells.
No, I mean, my main questions were, I think Marty really touched on it earlier, was kind of looking at what the applications of this will be possibly down the line. I know in your video, Kitch, on this, you mentioned things like, sunblock and stuff, which I guess was kind of out there, but yeah, what applications you could see coming from this technology further down the line?
Well, this will probably be this is more of another option to produce these nanoparticles. But there could be some side spin-offs. Sorry, I just noticed Enrico has gone offline when I've actually become what's the phrase? Convalent, or when I actually can talk. Typical, isn't it? But there are some side projects that could come out of this. For example, Enrico brought up bioremediation. That is the basically the removal of pollutants from the environment. One such thing we can actually do is increase the metal reduction rate, we can increase the metal removal rate from environmental samples. For example, there are some soils that can be contaminated with metals such as uranium, chromium, and wastewater runoff from gold and silver mines. You can put that into, we can use this technology to increase the removal rate of those compounds. Not compounds, metals. Let's see, Enrico is just dropped out. It's a Singapore connection. That's the one thing they don't have good there, is a good Wi-Fi.
Just looking through the chat, Microblogganism has asked a question: where would you naturally find Shionella?
Shionella you would find actually you would find Shionella in the ocean, in the sediments, in ocean sediments. It's a marine bacteria, so it's a bit of the universe, as actually point out. Shionella lohica is actually one of the organisms I'm working with. Was actually found off the coast of Hawaii. Not too sure if you have to start the call back because Enrico said he's back, but I'll just finish this anyway before I start the call again. And in fact, I think Shionella lohica, or some species of Shionella, are actually found in the intestinal tract of marine life. I'm just going to restart the call just to get him back. And there we go. Hello?
Hello?
Yes, sorry. Sorry, guys. This is. I can see.
Oh,
there we go.
am I back? Yes.
Yes, actually,
I don't know how long my releases work exposition was heard, but it's okay I'm okay with that.
Okay. Did you hear the question about where Chanel are usually found?
No, but I can answer. Chanel is usually found in sediment, particularly sediment at interface between aerobic and anaerobic zones. There have been Chanel found in Antarctica, Mississippi, Missouri, South America, in Argentina, in various lake sediment. North America in Lake Oneida, the first they were found. Some of them are discovered in Southeast Asia and so on. I think about 40 locations where Chanel was found so far, including the deep sea vent and 5,000 meter sediment.
And just at this point, I just like to remind people that the link to donate to the experiment.com page for this brilliant, beautiful work is actually in the channel description. It's the first link. It'll be experiment.com project page. So if you can donate anything, or if you're willing and able to donate, please do. It's just in the channel description. So actually, I'm not sure if this is a question or not, but I had one here. Now get pumped, link to channel. There we go. I see this is from Trollsar. I see you're using it as something to build non-organic electrical films or particles. Those are you're sizing nanoparticles, metal nanoparticles, particularly silver, though we're hoping that this will be used as a model to build on other nanoparticles, or to synthesize new other nanoparticles such as gold, palladium, maybe even if we can get it to that stage, we could maybe even look at bimetallic nanoparticles. Definitely not during the course of my PhD, but maybe on the further down the line by another PhD who's maybe interested in this.
It's basically what I guess I'm wondering about is, as I understood it, the nanoparticles were supposed to be used for in medical treatments. Or is it just like you don't really care what they're used for, you just want to know how to make them? Or
at the moment we're more concerned with the synthesis and then finding the application, because at the moment that's what my PhD is mainly concerned about. But there are applications for silver nanoparticles already being looked into, such as silver is a very good antimicrobial agent. Silver can be used for drug delivery and also for catalytic purposes, in order to speed up slow down reactions. But silver is not the only metal we will be interested in, or if someone else takes on this project after I'm finished with it. Silver may not be the only metal that can be used in this process. We could also use other metals such as gold or palladium, which have their own applications. Gold is actually quite a fantastic metal because it's not toxic, but it's not toxic, but can have a wide range of applications, such as drug delivery. We can use it to deliver drugs in the body. You can use it in the treatment of tumors. It has applications in electronics, which I'm not really too familiar with, and wouldn't be too familiar with the electronic purposes of these gold nanoparticles. And it can also be used in sensors in order to detect bacterial DNA or tumor cells or other antigens.
Michael,
yeah.
Let me interject a moment.
Sure.
Going to the question of the drug delivery. When doing drug delivery, it's quite important to have a reproducible material, in this case nanoparticles, so that we attach our drug with a very well-defined geometry, and the drug kinetic is the same from batch to batch. This is usually obtained through a very complex standardization process in chemical nanoparticle production. This is probably the most expensive part, and we think we can save some of this hassle by producing nanoparticles with much more focused distribution using bacteria. So the main application will be drug delivery or direct application to biofilm, infection, cleaning of linen in the hospital, and so on. However, prior to this application, which are very interesting but are still far from us, we need to define, which we need to understand which is the best way of producing a nanoparticle with good side distribution. And that's exactly what the PhD of Michael is about. Once we fix this part and we understand how to produce nanoparticle with the defined side distribution, we can then start investigating the application. And Michael is collaborating with Paul Cahill at the CU, which is much more interested in the downstream application, and will take the work from there.
Okay, I actually got a couple of questions from chat. This one from Microblogganasm. So you were using electricity to increase the number of electrons available to the bacteria's enzymes.
Can I answer this question?
Sure.
Yeah, that's about the correct answer, but not only the number, also the energy of the electrons. Basically, by providing additional potential, we are providing an external electrochemical driving force, which increase the energy of the electrons already delivered by bacteria through their enzymatic chains, and so the reaction is faster and result in the production of smaller nanoparticles.
Mm-hmm. And there's a second question from A Bit of the Universe. How do you control the electricity on such small scales? Is this on the scale of transistors in a modern CPU? Is the biofilm coated on something larger?
Well, that's an interesting question. At this point, we cannot control the electricity distribution, the potential distribution on a microscopic scale. Our experiment work with an average system, which is an electrode few square centimeter wide, and we only apply potential to the whole surface, and we don't care so much about the potential distribution. This potential distribution on the electrode definitely will alter the production of nanoparticle in the system. Ideally, we should have a flat microfluidic system where the potential is applied in a much more homogeneous way, and nanoparticles are produced from the entrance to the exit of this microfluidic cell. This is quite difficult to scale up, and it's also quite difficult to set up for the moment. So we start with this simplified system. Biofilm is actually naturally developed on the surface. We are using carbon felt and other material because of the large surface and because of the bioavailability characteristic. We can also use other more defined surface like silicon, gold, or other bulk material. However, in this case, the current observed in the nanoparticle production is so small that we cannot go into further characterization of nanoparticle through microscopy or spectroscopic method.
Okay, just going through the chat.
I hope this answered this question.
Yeah, I think so. Do you guys, do you have any other questions?
And cells extract, because reducing enzyme either in vivo or in extract are able to interact directly with the metal ions and reduce them to the zero valent form where they precipitate as nanoparticle or various form of aggregates. This is just fine. However, there are two problems associated with this biological reduction process and formation of nanoparticle. The first problem is this side distribution, which is very broad and is not useful for further application and for downstream processing. The second problem is the long time required for biosynthesis. Early experiment using fungal and bacterial biomass proved that using viable biomass, the reduction reaction occur in 24 to 72 hours, which is way too long for industrial application. Using using cell extract, the reaction time goes down up to 20 or 60 minutes. However, cell extract is expensive because it is not more viable cell, so it cannot replicate, and they need to be substituted every time. So the key idea is that bacteria are able to interact with electrodes. So we can probably provide a bit of extra energy in the form of applied potential to facilitate the metal reduction process, which occur either extracellularly or on the cell membrane. That's the idea that we started to explore with Michael in Singapore in 2012. We have currently some draft in preparation, and that's why we try to push down further this idea on the experiment.com platform and prepare a project that Michael is now supporting. Any other? Can you take it from here, Michael? And then.
Sure. The experiments to date so far, we've looked at the effect of the bacteria itself. We're looking at Shigella onidensis as compared to Shigella lohica. Shigella onidensis is actually the model organism at the moment, the model organism for bioelectricity, at least in terms of Shigella. But Shigella lohica actually has a faster metabolism. Therefore, we thought that it would be interesting to compare the two in terms of see if there's actually much of a difference in their performance for the nanoparticle production in the electrochemical cell. We're also looking at the electrode material, which can be an overlooked parameter.
Michael, sorry to interrupt you. Maybe we should outline better for the listeners how this process works. You have
yes, please, because I don't understand a damn thing.
Yes, good. Thanks for the feedback, guys. So, Michael, want to start describing how you set up on this experiment, and then tell basically which is the biological process happening, and then we go into further details.
Sure. Well, first of all, we set up a three electrode electrochemical cell. Now, there's as the name implies, there are three electrodes, obviously. The first one is the working electrode. This is where the magic happens, where we grow a biofilm and where we apply a set potential for about 48 hours, and which allows the biofilm to form. It uses the actual working electrode, similar to how we use oxygen, as an electron acceptor. There is a counter electrode there that completes the circuit in the electrochemical cell. And there's a reference electrode. This allows the potentiostat, which is the controller of the system, to basically gauge it's a point of reference for the potentiostat that allows the working, so it can fix the potential of the working electrode.
Okay, but what is the biological process happening here? You want to talk about, or should I a moment?
The biological process, well, our hypothesis is at the moment is that once the biofilm is formed at the electrode, the working electrode, we can then charge the working electrode at a set potential for about 100 minutes or maybe even less. And basically the electrons, we assumed or hypothesized is that the electrons hop from the working electrode to this structure on the outer membrane on the bacterial cell surface, which then is transferred to the silver ions. Now we need to do a lot more experiments to actually demonstrate that this model is correct, and it may not be, but we're just at the very early stages of the actual mechanistic model. Maybe if there's anything else I might have missed out, or...
Is working for this with the system since 2 years and a half, so probably tend to assume a lot of things because it is the practical expert of the system. Let's back up a moment and try to make an analogy with the human respiration. Okay, we are all familiar with the process of respiration. Basically, we use oxygen. Sugar, glucose, and reduce oxygen to conserve the energy that in this process allow the maintenance of homeostasis and ultimately our life. Okay, I'm doing very simplistic summary here. Bacteria do something similar. They are able to respire either aerobically or anaerobically by reducing sorry, and transferring part of the electrons
you're cutting in and out there, Enrico.
The process of aerobic and anaerobic respiration, where oxidation of a substrate like glucose and acetate in bacteria result into the reduction of a terminal electron acceptor. Can be oxygen in case of aerobic respiration, metal or soluble redox acceptor in case of anaerobic respiration.
Sorry, sorry, you can't.
That again?
Yeah, sorry, but I can summarize just what you just said there.
Please.
For human cells, for usually for cells such as our cells in our body, we take glucose as sugar and convert that into energy through respiration. Now, what happens is that we basically start to break down glucose, and which releases electrons, high energy electrons, which then can be used to form what is known as ATP, which is an energy carrier. But through this, you lose energy and give it to the ATP, and at the end we need to get rid of that electron. Otherwise a respiration will stop and we die. That's not good. So basically that is the role of oxygen. Oxygen comes into the body, comes into the cell to the mitochondria, and is then accepts the electron and hydrogens and two hydrogen ions to form water.
Okay. So what's happened then with bacteria, which is the analogy between bacterial respiration and cell respiration? That's where our starting point.
Yes. In the analogy with that transfers to bacteria as in an anaerobic environment. That means without oxygen, it obviously cannot use oxygen, so it's in trouble now. So basically, it has evolved this little ingenious little system where it can actually transfer these electrons to other electron acceptors other than oxygen, something we don't have that ability to do. So it can transfer it to metals such as iron oxides, gold, silver, even quite nasty little metals such as uranium and plutonium. This is what that Shrinella has done. Shrinella is not the only bacteria to do this. That's interesting enough. Geobacter can also do this process. However, we tended not to use Geobacter because if it's exposed to oxygen at all, it's going to die, which raises the cost of the whole synthesis and will basically raise the cost of our own lab, and it's quite finicky and not very appealing.
Okay, so which is the innovation of the project that you're proposing, Michael?
The innovation is that it combines the benefits of the biological methods, which are that the nanoparticle, the method is quite user-friendly. That means that each batch is more likely to be consistent, as opposed to processes which can be more complex, and then can lead to differences in the nanoparticles produced. The time is also drastically decreased. For Shrinella, it's 48 hours, 2 days, to 100 minutes, and sorry, there is one more. And the size distribution can be quite narrow, which is actually something we're working on at the moment to try to further decrease in order to match the conventional methods.
Okay, but why? You say that in your experiment project that I'm reading right now, even if I wrote with you, it's okay. You said that you are applying a potential, meaning electrochemical potential to these cells. Why is it important to do that, and which is the resulting innovation in this process?
The electrical potential actually is the stimulus. The electrical potential actually allows for an increase in the metal reduction rate. So that means that we're transferring electrons to the metal ions at a much faster rate. this means that the synthesis time decreases and the size distribution of the nanoparticles will also decrease because of the increased focus.
So basically what's happening is that by applying a potential, we are providing an external contribution of energy.
Yes.
And since this is a catalytic reaction that results in the creation of a nanoparticle, we are facilitating this reaction by adding energy. This can on one side decrease the activation barrier needed for the reaction, which in the case of silver nanoparticle is fairly small. On the other side can increase the rate. Increasing the rate of nanoparticle formation has an interesting effect on the size distribution. We tend to have a smaller nanoparticle and producing much faster. Instead, on the other hand, natural, quote-unquote, production of nanoparticle results in a wider size distribution and much slower process because the goal of bacteria is not to produce a nanoparticle but just to survive. So with potential, we are providing an additional help for this process. And more interestingly, by changing potential, we might, in the future, be able to determine the size distribution. So ideally, we should tune the potential applied to obtain the distribution that we want for our specific process, be it a drug delivery process or a nanomaterial production process.
Okay. And with nanoparticles, the smaller size is better, especially for nanoparticles like silver. Is there any questions from maybe our other hosts?
Well, I don't really know where to start because I haven't really understood all that much of this.